new meeting. Dear colleagues, dear attendee, and dear friends, first of all, do you hear me? And secondly, it's a pleasure and it's also an honor to welcome you to the 11th Symposium of the Society for Research on the Cerebellum and Ataxia. And it will be a virtual symposium, but it does exist at least. And to make a long story short, we were used now in this society, it's a small but active society, to have a symposium every year. And last year, there was supposed to be a so-called 11 international symposium. And Jan Sendelin from Pilsen University, from Pilsen and Charles University proposed to organize it. And we accept at the board, and he has been working very hard all together with the scientific chair from the association, with Tim Moti Ebner, and they propose a program, and then a small event occurred, a world pandemia. And of course, this pandemia, that is one of the most terrible things that could occur in the last years, obliged us to cancel or at least to postpone the meeting because we didn't know the situation. There were several waves. The situation was different in different countries. So it was impossible to organize it. So we decided to postpone it. And so we have now, again, the 11th International Symposium, and Jan and the team work on the new program or some modification in the program. And I am happy that this has been done. And of course, it's a virtual meeting. And the virtual meeting, we are all used now to Zoom and Teams meeting. And I realized by working and with the society Semico and Luc Niville that the organization of the virtual meeting is much more complex than a usual Team or Zoom conference that we are used to do now. And so Luc, that I want to thank already, has organized a quite complex but easy to use organization with a live lecture, pre-record lecture, questions, etc. that you will see. And I am happy that this will occur now because as I told you, our society is small but very active. And another feature of this society is we try both to do excellent science and to have a kind of, at least what I found, friendly atmosphere. And this is very nice. And we want to maintain this atmosphere and the excellence of science at the same moment. A virtual meeting, in my opinion, will never replace a true meeting because we miss something like many things like chatting with friends, looking at really posters, spending time in poster, drinking, chatting, etc., etc., the real life. And we do hope that next year we will be able to have a new meeting in a real life and not a virtual one, although a virtual is much better than nothing. And so I think now we will have, during this opening ceremony, a small talk from Tim Ebner. We will present the program and also a talk from Jan that is that has been organizing with his team the meeting. The meeting is named from Jan Purkinje to the future. So I would say it's from Jan Purkinje to Jan Sendelin and to the future. And it's very important to understand that we are both in the tradition of the excellent science of these big names in the field, but we want to be also closely linked to the future because the function of the cerebellum appears to be increasingly diverse. And this will be illustrated, I think, in the Ferdinando Rossi lecture. So Tim, if you want to speak, I will stop my.
Thank you, Jean. I'm going to put up the program. I hope you can all see that. But before I go over the program, I just wanted to add a couple comments to Jean's introduction. I think the Society for Research on the Cerebellum and Ataxia is in a very unique position among cerebellar societies. It really is the one that embraces both basic science through cerebellar disorders and therapies. I've been involved in a number of organizing a number of these. I think actually in all 11 now, but I would say that what we've seen is enormous progress in understanding the cerebellum. And I think nothing highlights that progress more, this progress over the last decade, than the fact that in this 11th meeting, a large number of the talks are dedicated to actually therapy of cerebellar disorders. And that certainly wasn't the case 11 years ago. To review the program, again, in a little bit, we'll have the Fernando Rossi lecture by Jeremy Shaman. There'll be a quick break. After that, we'll have the first session on pathophysiology and cerebellar disorders. That will be followed by a general discussion. And during that general discussion, we will also review 10 pre-recorded lectures. After that, today will close. Now, I want to also emphasize that there are some 30 posters that we hope you will view. Tomorrow, again, we begin early in the morning, at least in Minneapolis. The second session will focus on cerebellar function and development. That'll be followed by a third session on dystonia, the general discussion session. In that, on the second day, we'll it has 8 pre-recorded lectures. Presently, I think we will have 7 of those do their short presentations, and followed by the closing ceremony. At that time, I think that'll be important because Professor Zhu from Nanjing will present the next meeting at his university, the University of Nanjing. Again, please note that in both days, there is a period after almost 2 hours of open discussion for free and discussion about the talks or anything you would like. And then lastly, I do have to again state that I have to thank Jan and the organizing committee and also Luc Neville putting together symposiums. International symposiums in particular is very hard work, but I think we found this particularly challenging to do a virtual symposium that spans the entire world. And again, I want to in advance thank all the speakers and poster presenters. And I hope you enjoyed the meeting, and I'll turn it over to Jan.
Thank you. Dear colleagues, ladies and gentlemen, let me also welcome you to the 11th International Symposium of the Society for Research on the Cerebellum and Ataxias. As Professor Mariani already mentioned, it has been planned in Pilsen last year, but due to COVID pandemic, it has been cancelled because traveling was almost impossible. And now the situation is hopefully improving a bit, but still not safe, and any predictions are difficult, and it still wouldn't be responsible and safe to make a classical on-site meeting. So the society decided to organize a virtual meeting. As Professor Mariani said, there are some substantial disadvantages of such a virtual meeting. We cannot meet in person. The interactivity of such meeting is also disadvantage, and during the coffee breaks, we can offer you only virtual coffee. But the advantage is that the lectures have been or will be recorded, and you can see them as well as the posters during the next 1 year, because for 12 months the web page of the webinar will be maintained and you can see all the lectures again and again. I will, or I hope, here is the realization team. So the meeting is organized as a meeting of the Society for Research on the Cerebellum and Ataxias, represented by the editor, by the executive committee, by the board. Professor Ebner was the head of the program committee of this meeting. The organization team comes from Faculty of Medicine in Pilsen, Charles University, and I would like to thank the leadership of the faculty to enable joining this project. My colleague, Dr. Alena Zumrova, is a member of the second Faculty of Medicine in Prague of the same university, and Professor Asif Shaikh, the organizer of the dystonia session, comes from University Hospital Cleveland Medical Center in the USA. The important partner and member of the realization team is Semico and Luc Niville and his colleagues, who provided all the technical and administrative and organization support to the meeting. And I would like to thank to Springer, the general sponsor of the conference, who supports the meeting. I hope you have all seen the instructional video that is available from the homepage of the webinar. So I can remind just the main points how the meeting works. Now we are in the live part of the meeting, particularly now today in this virtual room. Tomorrow I ask you to enter this room. After the meeting, you will find here in these two virtual rooms records of all the live lectures presented during these 2 days. Then, after finishing the official program on both days, you can also enter the discussion room, which will be open for 2 hours every day, and everybody can activate his or her camera and microphone and answer questions. It could be indeed a bit chaotic, so I would like to ask you to switch off your microphones when not speaking, to reduce noise level. Where this image, this icon, you can enter the section where you can find all the pre-recorded lectures. You can read the abstracts and contact the speakers if you have some questions. Here we have a similar section for posters. And now a couple of words on how the discussion will be organized, because discussion is a very important part of each conference. Authors of the posters can be contacted via email, just clicking this icon next to each poster. And I would like to ask the authors of the posters to answer all these questions. The speakers of pre-recorded keynote lectures can be also contacted via email using the same button next to the record of the lecture on the page. But there will be also general discussion sessions, as Professor Ebner mentioned, 10 lectures today and 8 or maybe 7 tomorrow. You can find the lectures included in this or that session in the program. For asking the questions, use please Q&A module, type your question, and the chair of the session will select the most interesting, most relevant questions and present them to the speaker who will answer. There might be maybe not enough time to answer all the questions, but you can vote for the most interesting question to upgrade it, to highlight it in the list of questions and increase the chance that the chair of the session will select it. Live keynote lectures will be followed immediately, always by 10-minute discussion. You will ask again via the Q&A module, so the principle is the same. And finally, there are the two days or two free discussion rooms today and tomorrow for 2 hours each day after finishing the program. So if you want, you can join them to continue in discussions. So thank you for your attention. Dear delegates, enjoy the meeting, and within a very short break, within 5 minutes, let's start the scientific program of the conference, the Ferdinando Rossis lecture. Thank you. Dear colleagues, ladies and gentlemen, the scientific program of the meeting of the Society for Research on the Cerebellum and Ataxias will be traditionally opened with the Ferdinando Rossis lecture. It is a great honor and pleasure for me to welcome the speaker for this year, who is Professor Jeremy D. Schmehmann. Let me briefly introduce him, even though you all know him well. Jeremy Schmehmann is a medical doctor and a professor of neurology at Harvard Medical School and Massachusetts General Hospital. He is the founding director of the Ataxia Unit, director of the Laboratory for Neuroanatomy and Cerebral Neurobiology, and a member of the Cognitive Behavioral Neurology Unit. Professor Schmehmann's research is focused on the anatomical substrates of intellect and emotion, with a particular focus on the ataxias and other cerebral disorders. He's the author of the term cerebellar cognitive affective syndrome. He has defined and described, and the syndrome we nowadays know also as the Schmehmann syndrome. He is also an active member of the Society for Research on the Cerebellum and Ataxias. He works in the executive committee as the treasurer. Now Professor Schmehmann will give the Ferdinando Rossis lecture entitled Cerebellar Cognitive Affective Syndrome, Implications and Future Directions. Professor Schmehmann, please activate your camera. Thank you. And the floor is yours, please.
Well, thank you very much, Jan. Thank you for your generous introduction. Welcome to everybody. Wonderful to be here. I'm thrilled to be here in the capacity as the Ferdinando Rossi lecturer this morning to the SRCA. This is truly an enormous honor. I'm thrilled to have this opportunity. I'm very grateful for it. And I'm humbled to join the ranks of the esteemed awardees who have given this prestigious lecture over the years. So this is a great honor, and thank you so much. My topic this morning is the cerebellar cognitive affective syndrome, and I'll be presenting the story of how we got to this point. I will start with just disclosures. I'll be mentioning some of these books and a couple of the scales that we have devised over the years. I think I'd like to start by framing the story with a patient in the tradition of neurological grand rounds. And I'll tell you about this youngster whom I met at the age of 12. She was finally subjected to an MRI age 4 after an early childhood of behavioral problems, global delays in intellectual function, particularly with speech, language, and learning. And at MRI, she was found to have a tumor, which turned out to be a pilocytic astrocytoma. Now, she had the surgical procedure, the tumor was removed, and the location of the surgical site was through the midline vermis, and you can see the scarring here, which on the atlas map shows that the approach was through the posterior vermis, lobule 9 in particular, and it would have influenced the adjacent nuclei, the vestigial in particular. Now, what has been going on with this little girl through time is a range of behavioral changes that have received a number of diagnoses. Her emotional lability and mood swings and inappropriate giddiness, impulsivity, executive dysfunction, impaired working memory, mathematical skills, verbal fluency impairments, and perseveration all led to a variety of diagnoses: obsessive compulsive disorder, bipolar disorder, attention deficit hyperactivity disorder, and autism. Interestingly, when she had a motor examination on the brief ataxia rating scale, she was essentially normal, a little trouble perhaps with tandem, a little trouble with an arm, and some saccadic pursuit. Otherwise, this was not the issue for her. But her testing showed difficulties on full-scale IQ, verbal comprehension, spatial cognition, reasoning, working memory, processing speed, and difficulties with scores on attention and executive function. She had a neuropsychiatric rating scale, which is still in the preliminary form, completed by her parents, and she had a number of high abnormal scores on attentional control, emotional control, autism spectrum, psychosis spectrum, and an evaluation of social skill set. So this is a youngster who sets the stage for us of a cerebellar tumor resected, showing essentially normal motor examination, but a range of cognitive and neuropsychiatric impairments. Now, we learned through medical school and graduate school that the cerebellar clinical syndrome was ataxia, dysarthria, dysmetria, and oculomotor abnormalities. And we learned this from our great teachers over the years who identified how to perform the cerebellar motor examination. But I'll leave you at the end of this, I hope, with a sense that this is in fact the cerebellar motor syndrome because there's so much else that goes on in the cerebellum. And the cerebellar motor phenomenology dates back to Marie Jean-Pierre Florenz, who in 1824 embarked on experimental investigations in pigeons, a major undertaking at the time. And he showed that the cerebellum was critical for coordination. Unlike Rolando, who thought that the cerebellum was important for actual movement and power, Florenz's conclusion was that the cerebellum was important for coordination because the birds could move their wings, but they were flapping in an erratic fashion and couldn't fly. So the cerebellum from that point on became the motor control part of the brain. And Florenz also championed equipotentiality, that the cerebellum could do every function throughout. There was no particular localization of function within the cerebellum. When I was a resident in neurology in the neurological unit of Boston City Hospital, I met these two patients who set me on my path. One had a stroke in the genu of the internal capsule, and the other had a stroke in the caudate nucleus and adjacent region, both on the right side. And both of these patients had a hemi-neglect syndrome, difficulty copying a star, trouble drawing a person, a clock, missing out the left side of the page when circling the letter A, and very poor placement of cities on a rudimentary map of the US. Now, in the neurological unit, which had been led by Dr. Derek Denny Brown, who was one of the major figures in American neurology, the idea was that the basal ganglia were critical for their relationship to movement. And so when I met these patients, it raised for me the question, if the basal ganglia could be involved in non-motor control, what about the cerebellum, the big motor machine downstairs? Could that be involved in cognition? Well, I was following in some giant footsteps because Vincenzo Malacani, whose book I looked at when preparing the atlas, I'll describe it in a little while, studied the cerebellum, according to Zanata and colleagues, because he was interested in the neuropsychiatry of cerebellum and brain function. He wanted to figure out, was there a relationship between the number of folia in the cerebellum and intelligence? So the origins of contemporary cerebellar anatomy has its basis in the question of neuropsychiatry from Malacarne onwards. Throughout our training and contemporary neuroscience, we have understood the basis, the brain basis of behavior to be determined by the distributed neural systems in the cerebral hemispheres, each of which comprises these anatomic regions or nodes with unique architectural properties that work together in a coordinated fashion to enable behavior to emerge. The emergent properties of behavior arise from these interactions of the cerebral cortical areas. But there was no discussion at all of cerebellum. It was not present in the conversation. So when I came to the question, could cerebellum be involved in cognition, I was very fortunate to be able to meet, work with, and learn from my friend and colleague for many, many years, Dr. Pandya, who just passed away last year. For the purpose of studying the interactions at the anatomical level between cerebral hemispheres, association areas in particular, and the cerebellum through the pons. So we injected tracers, isotope tracers, and sometimes HRP, and we studied the tracts in the white matter as they coursed down through the cerebral peduncle into the basis pontis, because the cortico-pontocerebellar circuitry is a feed-forward limb of the cerebrocerebellar interaction before the projections feed back up into the cerebral cortex. So the first paper that came out of this discussion, and the question here was looking at the posterior parietal cortex, because the caudal parietal region is strongly interconnected with other associative and paralimbic regions of the cerebral cortex. And what we demonstrated was a very clear pattern of projections, not only from the superior parietal lobule, but from the inferior parietal lobule, including the caudal areas that are part of the associative paralimbic circuitry. And this led us to the conclusion that the cortico-pontocerebellar pathways permit a cerebellar contribution, not only to the coordination of movement, but also to the modulation and integration of higher function. So this allowed us to embark on this story, this journey of the anatomy of the circuitry that could support a cerebellar engagement in higher order function. And over the next number of years, through a series of studies, which I'll just summarize in one slide, we found out that with the injections of tracer in cortex and determination of terminations in pons, that there is a relationship between the sensory motor regions to the more caudal parts of the basis pontis on the ipsilateral side, but there are projections in a topographically precise way in a mosaic pattern of interconnections from prefrontal association regions, posterior parietal, superior temporal, parahippocampal gyrus, and parastriate regions into the pons. So these are regions important for executive function, attention, planning, working memory, strategy formation, spatial awareness, the valence, the limbic valence of spatial information, language processing, facial representation, and aspects of working memory. Around this time, we also were noting then that Dwayne Haynes had demonstrated connections to and from the hypothalamus to cerebellum. And so we had the story of the associative projections to the cerebellum and connections to the hypothalamus, which is critical for not just temperature regulation and blood pressure and heart rate, but important for a number of emotional, mood, and personality traits, as we have learned through the years, through the hypothalamic influence on behavior and its influence on other circuits in the limbic system. Now, we knew at the time that there were projections back from these cerebellar regions through thalamus into cerebral cortex, and so it led us to the view, encapsulated in 1991, 30 years ago now, that the cerebellum, in fact, was important in higher function, And this was an idea subserved by the concept of dysmetria of thought. And I conceptualize this as in the same way that the cerebellum regulates the rate, the rhythm, the force, and the accuracy of movements, it does the same thing for the speed, capacity, consistency, and appropriateness of mental or cognitive processes. So that was the idea. In a sense, that dysmetria of movement would be matched by an unpredictability and an illogic to social and societal interaction. There's very psychiatric terminology just embedded in that sentence. And the way that things would occur is that inability in the motor system to check the parameters of movement would be equated with a mismatch between reality and perceived reality and erratic attempts to correct errors of thought or behavior. I'll come back to this later. There are many ideas about what the actual transform would be, but this was the overarching concept of the dysmetria of thought theory. Now, where the anatomy comes in is we have these two competing realities. On the one hand, the mostly invariant architecture of the cerebellar cortex, which goes back to Ramon Cajal, which has been studied extensively and continues to be so now in the era of genetics. But this contrasts with the heterogeneity of the cerebellar connections with the extracerebellar structures that I was just talking about. And this became further elaborated upon with the transsynaptic retrograde and antegrade viral traces that Peter Strick has worked with so well through the years to show that the motor regions are talking to the dorsal dentate and interpositus, whereas the prefrontal regions are talking to the ventral dentate. And the motor cortex M1 is interacting with the anterior lobe and lobule 6, with lobule 8 as well as a second representation, which I'll come back to. And area 46 in the prefrontal cortex is interacting not with these motor regions, but with the lateral hemisphere, crust 2, and also down here in lobule 10. This becomes further investigated in imaging studies in humans, which I'll show. So this point brings us to the late 1980s, where we have the question of cerebellar cognition. We have anatomy that supports this, at least in the corticopontine projections, and the beginnings of the evaluation of the feedback system before the retrograde traces show the projections back in the work of Strick and colleagues. And the question that was raised was, well, if the cerebellum is involved in behavior, what kind of behavior? What is it that happens when a patient has a cerebellar injury in terms of the non-motor phenomenology? And so I had the fortune to meet this young woman who was 23 at the time, who had a fall on the ice. She had a CAT scan performed, and she was found to have a tumor. The tumor was resected. It was a midline cerebellar ganglioglioma, and she's still living and still dealing with some of her issues. And what we found at the bedside was that she had very little in the way of motor phenomena, but she had a change in personality. She had difficulty with her directing her behavior. She was disrobing in the corridor. She was eating with her mouth full, being rude to her parents, just a real change in the way that her personality was constructed. And in some simple bedside tests, she was perseverative when trying to bisect a line. She had difficulty drawing a clock and tried a few different times. And she had agrammatism. I am a young girl who's 23rd, and perseveration in trying to write a simple sentence. So she got us going, and we looked at a number of patients over the next few years. Predominantly with stroke. She was our proband. Stroke patients, those with post-infectious cereballitis, and a few with cerebellar cortical atrophy. And in these patients, at the bedside, just with simple testing that I learned from Norman Gershwin, one could identify difficulty with executive function, working memory, for example, perseveration, reasoning, calculations, and an affective change that occurred when the people have midline lesions. Visual spatial impairments, verbal memory, and language processing were all impaired at bedside testing. Visual memory, not so bad. Arousal, not too bad. Some problem with remote recall and apraxia, but these were the predominant problems just with the bedside testing. And Janet Sherman took these folks into the neuropsychology laboratory, and we identified on the Z plot, where 0 is normal, 1, 2, and 3 standard deviations below or above the mean, that there was a scattering. Some people were just fine, but some were pushed off to the left, for example, in the phonemic fluency task, or with trails, or with visual spatial function performing the ray copy, or with verbal and visual memory. And as a group, the patients with cerebellar diseases were pushed off to the left. There's an impairment in intellectual function. And here's a couple of examples of their performance. This is the copy of the ray diagram during the post-Epstein-Barr virus cereballitis. It's quite piecemeal and fragmented. There's no order to the way that this patient followed the instruction to copy the diagram. After we had treated her with plasmapheresis and she recovered, this is a nice orderly copying with the black and then the red and then the blue in a very structured way, which is different than she had when she was ill. This is not an ataxic dysgraphia, it's a poor planning. And this is an example of perseveration in a man with bilateral pica strokes and one with a right pica stroke. Copying of the two-loop diagram is markedly perseverative, like a frontal lobe patient. Here's impairment in copying the cube, star, and then frank perseveration in the Luria diagram, and a very rudimentary drawing of the person by a pharmacist who should have been able to do better than that. And so we conceptualize this as a cerebellar cognitive affective syndrome with impairments in executive function, spatial cognition, language deficits, and personality change. The planning, set shifting, verbal fluency, abstract reasoning, working memory were the hallmark of the executive dysfunction. Visual spatial organization and memory, which is a marker of a gestalt, a bigger picture view of life, is how the right hemisphere spatial cognition can be conceptualized. And the language deficits included agrammatism, aprosodia, and anomia. And the personality change occurred after the midline lesions with this blunting of affect, the disinhibition, and inappropriate behavior. And our dear friends, Mario Manto and Peter Marion, who we lost recently and still mourn daily, described this as the third cornerstone of clinical ataxiology, and were generous in coming up with the eponym. The monograph in 1997 coalesced the field of cerebellum and cognition with a number of authors, including those in the society now, contributing to the work at the time. Now, we replicated the demonstration of the cognitive affective syndrome in adults in children around the same time. And we showed that there was a problem with problem solving, failure to organize, verbal or visual spatial material, visual spatial impairments, impairments in expressive language, long latencies, poor initiation, brief responses, not able to elaborate their ideas, word finding trouble, and confrontation naming. And memory was impaired for stories that they had to give on their own, but better with multiple choice. And I'll flesh that out again in a second. And then the midline lesions produced this affective change, irritability, impulsivity, disinhibition, and lability. And here's an example of a youngster trying to copy, recall immediately, and then later this ray figure. And this is almost like a loosening of associations, as you would see in psychiatry. Fragments spotted around the page, but nothing tying it together. There's no binding of this concept. We have subsequently shown with Xavier Guel, who was a postdoc, Francisco also was a postdoc, both now moved on into the fields in neurology, that there are metalinguistic impairments in patients with cerebellar dysfunction. Now, metalinguistics is how you use language. It's to communicate, to share ideas, to understand each other. It's not a Broca's or a Wernicke's aphasia. It's the language for social context. And so ambiguity, inference, metaphor, how you express yourself in a grammatically and contextually appropriate manner. These were all impaired in our patients with cerebellar dysfunction. And similarly, theory of mind, how you understand someone else's mental state to be able to act in an appropriate manner in the social context, is impaired in patients with cerebellar disabilities. These are the mental processes needed to understand, generate, and regulate social behavior as measured by this reading the mind and the eyes task that was initially found by Baron Cohen and colleagues to be important in the autism spectrum patient population. We then fleshed out the affective component of the cerebellar cognitive affective syndrome by talking with patients and families, our own evaluations and reports of the families. And we conceptualized these behaviors as belonging to these 5 domains of neuropsychiatry: control of attentional processing, emotional control, autism spectrum disorders, psychosis spectrum disorders, and social skill set. And we viewed these as having features that were both excessive or diminished, exaggerated or decreased, almost like the positive and negative syndrome scale of schizophrenia, hyperactive or hypermetric, and hypoactive or hypermetric, reflecting in essence this cognitive or emotional dysmetria as part of the cerebellar cognitive affective syndrome. We went on to replicate the findings in a larger group, 77 patients with cerebellar disease, another with 36 with isolated disease, and then 41, 77 total, 36 isolated and 41 complex, and then a replication cohort of another 39 patients. And we demonstrated again the features of the cerebellar cognitive affective syndrome and came up with a scale that has now been translated into multiple languages to be able to detect those essential features of the syndrome that are present when the cerebellar cognitive and limbic regions are damaged. And I'll show you that in a moment. So this is available for use now as well. And then just published last week is this patient-reported outcome measure of ataxia, which will be useful for clinical studies in the ataxias. But I want to point out that of all the questions that patients told us are important, almost 20% of them are in the mental domain, the affective and the cognitive domain. So these are questions and aspects of behavior that patients volunteered to us as being part of their clinical phenomenology in the setting of a cerebellar ataxia syndrome, predominantly genetic. But it makes the point that patients are telling us about this, and we therefore need to listen. If you go back into the early child story now, not just the adults with diseases, there's some very compelling evidence that comes out of the little ones who've had hemorrhages either in utero or very early in life, the preterm children. And what Kathy Limperopoulos and her colleagues have shown through the years now and continue to show is that whereas there are in fact neurological abnormalities with motor delay and some functional limitations, there is an expressive and receptive language problem. There are behavioral and social impairments and autism scores that reach greater than 40% in the children in whom the pathology is isolated to the cerebellum with the cerebellar hemorrhage. Now, here's an extreme example of a cerebellar disruption. We think it's not likely genetic, more likely a result of some injury or trauma. The children who are born with basically no cerebellum, cerebellar agenesis, although that's a misnomer. And here with the Z-score turned on its side, in one case, a marked impairment in the overall intellectual function. The Ray copy figure is very poor. The learning is impaired, and the phonemic fluency task and the trails, alternating sequences task are all impaired here, many standard deviations below the mean. And so in these children who have the cerebellar agenesis, there is again the same story of executive impairment, spatial cognition, language impairment, and some have autistic-like stereotypical performances, obsessive rituals, difficulty understanding social cues. And for those who want to find it, this young man is interviewed on the radio in the National Public Radio in March of 2015. Now, still with the children, and this is a big problem in the tumors in childhood, the presence of the cerebellar cognitive affective syndrome and a pediatric postoperative cerebellar mutism syndrome now receiving attention by an international consortium across Europe, the US, and the Far East. The pediatric cerebellar mutism syndrome has a delay of a day or two after surgery, usually from midline tumor approach. There's hypotonia, oropharyngeal dysfunction, cerebellar motor syndrome, cerebellar cognitive affective syndrome, and brainstem dysfunction, sometimes including long tract signs and cranial neuropathies. So the issue of the pediatric postoperative cognitive affective syndrome and mutism is a real bear in the management of children with tumors and is receiving great attention. Very nice work from Aaron Bowes, who was at our place until he moved to Iowa, shows that an important location of the pathology in these people is in the midline, the vermis and the lobule 9 and 10, with its efferent through the superior cerebellar peduncle into thalamus, both the MD and also the anterior thalamic nuclei, which then spread diffusely through the prefrontal cortices and the associative and paralimbic regions from the anterior thalamic nuclear group. Now, one of the things that got in the way of our ability as a field to really define and understand cerebellar neurology was the fact that cerebellum was really a little orphan child in the field of cognitive neuroscience. So this was the atlas that was widely used when functional imaging became established as a new way to think about brain organization. And what the Telerak and Turner Atlas shows is all this anatomy in the cerebral hemispheres, but there's terra incognita down here in the cerebellum, nothing. So together with colleagues in Montreal, we devised and published the Cerebellar Atlas, both as a paper in 1999 and a book in 2000, that resurrected the lost cell nomenclature with the bulk nomenclature across 1 and 2, and replaced the old terminology that was confusing and conflicting with each other for the different hemispheric lobules. And we identified the fissures that segregated the different cerebellar lobules from each other. We demonstrated that they were continuous across the hemispheres. And we showed nicely in the cerebellum that the anterior lobe in front of the primary fissure is relatively small in comparison to the massive expansion of the posterior lobe in the human cerebellum. This allowed us to start looking at the function, structure, relationship in the cerebellum. And so with using ataxia rating scales and stroke, the old way of looking at how brain is wired up to do functions, we showed that a stroke in the anterior lobe here in the superior cerebellar artery produces an ataxia syndrome through the modified International Cooperative Ataxia Rating Scale. But a similar size stroke in the posterior lobe, in the posterior inferior cerebellar artery syndrome territory, produces no motor impairment, but instead produces the cognitive affective syndrome. And with a more detailed voxel-based lesion symptom mapping study with Catherine Studley, who you'll meet again in a moment, and the group in Boston, we showed that when patients have a stroke in the anterior lobe, lobules 4 and 5 extending into lobule 6, here in the coronal plane, here in the sagittal plane, these people, in fact, have the old-fashioned cerebellar motor syndrome. But when the stroke spares the anterior lobe and only gets the posterior lobe, lobules cross 1, cross 2, somewhat cross 1, although didn't have much cross 1 in these patients, and lobule 9 and 10 and lobule 8B, these patients have the cerebellar cognitive affective syndrome only. They do not have motor incapacity. So you can have a dichotomy, double dissociation of motor syndrome from the anterior lobe, cognitive affective syndrome from the posterior lobe. We then went on to look at the functional imaging of brain through task-based analysis, with the first meta-analysis in 2009, looking at cerebellar functions across different studies. Now, we had tried this 10 years earlier when the atlas was first developed, but this was before the era of the activation likelihood analysis software. And this allowed us to do this. So Catherine worked with us to show that in the meta-analysis, the motor task activation was in the anterior lobe and down here in lobule 8, language in lobule cross 1 and lobule 6, spatial representation on the cerebellum on the left side. So cerebellum on the right side here, talking to, so spatial is cerebral hemisphere right, cerebellum on the left, and midline. So left cerebellum and midline, working memory is bilateral. In a single case study, the first demonstration, and this elaborates further as you'll see in a moment, this is the first demonstration of multiple different tasks in a single individual in the scanner. We showed for the first time that in a single person, there was a similar kind of representation. The motor representation, primary and down in lobule 8, language processing in the cerebellum on the right in cross 1 and lobule 6, a spatial representation on the left and then the working memory, in this case, on the one side, but in the group results of 9 subjects together, we see it again represented bilaterally. So in a single individual, any of us who jumps in the scanner, we can have a brain activation that looks like this, and you'll see how this develops in a moment. The group with Johan Dietrichson, who developed the suit atlas based on our cerebellar MRI atlas in 2000 and 1999, along with Maeve King and others, had people scan multiple times with functional imaging, and they show that there is, as we showed earlier, but in a much more elaborate manner, the anterior lobe motor representation with second representation down hand lobule 8, but all of these other areas in the cerebellum, the vast expanse of the human cerebellum, is engaged in tasks such as planning, working memory, divided attention, narrative processing, verbal fluency, action observation, word comprehension, and autobiographical recall. So a number of different activations through different tasks, but in topographically precise ways, different from the cerebellar motor syndrome. You then move to resting state functional connectivity. And what Randy Buckner showed is that with these different networks in the cerebral hemispheres, you can identify a sensory motor network, a dorsal and ventral attention network, a frontoparietal network, and then the red is the default network, important for the idling brain, the creative brain, the internal processing brain. And then startlingly, if you reflect these networks into the cerebellum, you see that there is in fact an anterior lobe predominant sensory motor representation with another representation down in lobule 8, but the vast expanse of the human cerebellum has the networks represented in them that have nothing to do with sensory motor processing. It's a dorsal and ventral attention, frontoparietal, and the default mode networks represented in the cerebellum in this great expanse of the cerebellum that has evolved through evolution. Xavier Guell, working with John Gabrielli's group at MIT, took this one step further. We build upon Snyder's work in the 1940s and 50s that set to rest the equipotentiality idea that Florenz had, that cerebellum does the same thing everywhere and nothing is differentiated. Snyder showed the topography of the legs and the arms and the second representation down here. It was Woolsey's term, second representation for lobule 8, with the visual and auditory inputs into posterior vermis. Xavier was able to show that with task activation and resting state, you can replicate the primary and secondary motor representation as we have here from Snyder. But in the cognitive representation, there is both through task activation and resting state. Buckner showed this in resting state that there's a primary, secondary, and tertiary representation. Xavier showed this for resting state and for task activation in the Human Connector and Project dataset of many, many hundreds of patients, and now over 1,000. So for language, working memory, social cognition, and emotional processing, which has a predominantly midline representation, there are primary, secondary, and tertiary representations of cognition in the cerebellum, not in motor-related areas. And you can take this even further. In the gradient network analysis of the strength of connectivity across the hemispheres, there is a dichotomy of motor on the one end of the spectrum to default mode processing on the other, with the maximization in crus 1 and crus 2 of lobule 6A. And you can map these in gradient space where you go from motor representation to task positive, where you're doing things, to task negative default mode network in different locations of gradient space, which I think will be very helpful as you look at functional imaging going forwards in locating where these activation pixels are in this kind of gradient space. So the sophistication has become increasingly elaborate as we understand the organization of non-motor areas in the cerebellum. And then just recently, we learned that in addition to the network mapping of cerebral hemisphere to cerebellum at the multiple individual level, you can map this at the individual scale. These are single people who jumped in the scanner many times. And what Buckner's group shows is that the reflection of the sensory motor network in the anterior lobe and secondary representation in lobule 8 is maintained along with a default mode and dorsal attention, ventral attention, salience, language, visual processing, all in the cerebellar posterior lobe, along a similarity. There's a similarity between subjects, but there are differences between individuals. And so the same spot in a particular folium may not have the same network mapping to it from subject 2 to subject 1. And this becomes important and kind of expected, as there should be individual differences in the way the brain is organized. This reflects down to the dentate nucleus, as one would expect. We know, for example, that when the dentate is involved, the recovery of motor function or the recovery of cognitive function is further delayed, and there are papers that demonstrate that, including our own. And we know from the connectivity of all the work that's been done in the beautiful anatomy through the years of the tight corticonuclear microcomplex that the nuclei are tightly, intricately interconnected with different regions of the cerebral cortex. Now, you can use diffusion imaging and you can show that the different cerebellar lobules are linked with different parts of the deep nuclei. And you can use resting state connectivity to show that there are different functional territories within the deep nuclei themselves. So there's a default mode network, a sensory motor and salience network, and a visual association network, all mapped within the deep cerebellar nuclei. This is a first pass attempt of this just from Xavier and the rest of our group last year, showing the anatomy, not only of the cerebral cortical organization, but the deep nuclei as well, along these motor to cognitive gradients. Just in the last few years, there's been some really very nice work looking at other aspects of the anatomical connectivity of systems that are beyond motor control. And I'll start with the ventral tegmental area mesolimbic dopaminergic system. We've known that the ventral tegmental area, which is the origin of the dopaminergic projections throughout the hemispheres, anterior cingulate cortex, prefrontal cortex, the bed nucleus, the stria terminalis, these are all very limbically relevant parts of the brain circuitry. The VTA has a connection from the fastigial nucleus. This goes back a long time. But what Cameron Kodaka showed is that if you modulate the deep nuclei through optogenetic activation, you affect the VTA and you change the behavior of the mouse. So now you have a functionally relevant pathway from cerebellar deep nuclei, particularly fastigial, to the VTA that changes behavior. And indeed, what the Wagner group shows is that the cerebellar granule cells encode the expectation of reward. So now you have a completely new understanding of what's happening in terms of the limbic valence of behavior and its role related to cerebellum. And then most recently, this beautiful work from Sasha Dulac and her group showing that the fastigial nucleus, which I've long talked about as having multiple pieces to it, it's important for balance, for oculomotor control, for limbic valence, for arousal, for autonomic function, indeed has separate different nuclear groups within it that can be defined by the genetic pattern and the genetic signatures as being linked to different parts of the olive, different parts of cerebellar cortex, with a postural motor piece, an oral motor piece, a vestibular autonomic piece, an orienting piece, and an arousal, including, for example, salience, behavioral switching, vigilance, motivation, and working memory, and so on. And the fastigial nucleus, therefore, becomes a really important player in the limbic cognitive dichotomy that becomes readily apparent both from the older work with fastigial nucleus stimulation in the animal models looking at behavioral changes, and the more recent work of lesion mapping showing that brain lesions involving midline cerebellum, including fastigial nucleus, change behavior in a profound manner. This has a reflection in some of the most pervasive diseases that currently in neuropsychiatry, including autism. So Bowman and Kemper, who were at Boston City Hospital when I was training, were some of the earliest supporters of my hypothesis about cerebellar cognition, because they were finding already back in the early 1980s that in the patients with autism, the deep nuclei were quite atrophic compared to healthy controls. And there was some loss of volume and Purkinje neurons in the posterior cerebellum. So now you have the deep nuclei involved in some of the limbic circuitry, certainly vestigial nucleus, and the posterior lobe now we know to be involved in cognition. And more recently, Catherine Studley, who's gone on to American University, shows that there are connections between, firstly, that there's an anatomical circuitry here in CRUS1 in the cerebellum and lobule 7 that is volumetrically depleted in people with autism through imaging studies, and that there's an abnormal connectivity between CRUS1 and the infraparietal lobule in autism. And there's a nice mouse model of the tuberous sclerosis complex mouse of autism that shows a similar kind of a story and is proving to be important in linking cerebellum to autism behavior and potential implications for therapy. And the schizophrenia, which you'll be hearing about from the speakers in this course and in the symposium, schizophrenia has a long story going back to the 1970s and 80s of linking smaller cerebellum, larger fourth ventricle, loss of a normal asymmetry, all associated with schizophrenia. And what we now have come to show as a group with Roscoe Brady and Mark Helko leading the charge is that when you do brain stimulation to the posterior vermis, which I'll show you in a moment, you change the dorsolateral prefrontal cortex connectivity in schizophrenia patients. And this change in the connectivity is related to an improvement in the positive and negative syndrome scale of schizophrenia. So cerebellar stimulation, which we did first in 2010 as a proof of principle in patients with schizophrenia, the network analysis of the vermis to the dorsolateral prefrontal cortex, which superimposes upon the work I showed you from resting state connectivity, has a direct implication in terms of cerebellar modulation in a psychiatric disease, causally related, and showing improvement in the clinical phenomenology. And here's the work that set this up, where if you stimulate the cerebellum through transcranial magnetic stimulation, we change the dorsal attention network by the cerebellar vermis stimulation, whereas if you'd go laterally in CRUS1 and CRUS2, you change the default mode network in the posterior parietal cortex, retrosplenial cortex, and the temporal lobe. And then what Faranak-Farzan showed is that using the EEG, you change these different networks, but in the same direction. And what you're doing is you're changing the temporal dynamics of these networks. Here's EEG patterning, which the changes in a similar direction, but in different locations. Vermis does the dorsal attention network, and CRUS1-2 does the default mode network, and different EEG patterning here. So this was the first proof of principle empiric analysis of the dysmetria of thought theory, that you are changing the networks in different places, but in the same direction. And I'll come back to that in a second. So here's the paper from 2010, where we took the idea about the cerebellar cortical interconnectivity in the hope that one could change cerebellar modulation of these upstairs networks and produce change in behavior. We stimulated the vermis in patients with schizophrenia as a proof of principle and a safety study. And we show improvement in the negative component of the PANS, improvement in depression, in happiness, sadness, and alertness, and in the attention task, the continuous performance task. This has been replicated by others as well, and seems to be a potential avenue for therapy going forwards. So let me finish then just with the concepts about where this is going and how we can think about this. Firstly, the dysmetria of thought theory, now 30 years on, is that the cerebellum is an integral node in the distributed neural circuits observing sensory, motor, cognitive, autonomic, and affective processing. And the key is that the cerebellar cortex is anatomically homogeneous by and large, but the different cerebellar regions modulate different functional domains. There's a functional topography, a sensory motor region in the anterior lobe, adjacent lobule 6 and lobule 8, and a cognitive limbic region in the posterior lobe, where the limbic circuitry is a vestigial nucleus and midline and lobule 9 and 10, which is also autonomic and vestibular, as you know. And the cognitive regions are the massive expansion of the cerebral hemispheres, the lobule 6, CRUS1, CRUS2, and part of lobule 8 as well. Now, the idea is that there is a universal cerebellar transform by virtue of the essential homogeneity of the cortex. Our view is that the cerebellum makes things better, optimizes performance by modulating behavior, whether it's motor or cognitive, around a homeostatic baseline, automatically, implicitly, and according to context. There are many investigators through the years whose work goes into that sentence. Context dependency, for example, Tom Thatch, automaticity, implicit learning, the modulation of behavior, all of the work that goes on at the level of molecular chemistry, the neurophysiology, the genetics, all of the work that people in this society and elsewhere do about how cerebellum functions has relevance for what the cerebellum is doing, not only in cognitive and limbic performance, but certainly also for motor and vestibular and autonomic. The idea then is that the cerebellum breaks, it breaks the same way. So if there's a universal cerebellar transform, there should be a universal cerebellar impairment, and that impairment is dysmetria. If you lesion the sensory motor cerebellum, you get dysmetria of movement. That's a cerebellar motor syndrome that we've known about since Florenz. But if you lesion the cognitive limbic cerebellum, you get dysmetria of thought and emotion, and that is a cerebellar cognitive affective syndrome. And so the new reality is that the cerebellar role in nervous system function certainly is ancient, goes right back to the beginning. That transform, the universal cerebellar transform, is necessary. It's evolved in concert with progressive elaboration of the cerebral association areas. There's a mosaic evolution of ideas, places that in the brain that are interconnected, evolved in concert with each other. Now to subserve higher function and mental health, in addition to sensory motor processing, autonomic function, and limbic function. And the cerebellum does for cognition and emotion what it does for motor control. That's a dysmetria of thought theory. So the challenge then is that we have this paradigm shift in our thinking and in the knowledge about the cerebellar structure function correlation. And this essentially invites a recontextualization of the way we think about the etiology and the therapy of disorders of mental health. If the cerebellum is important for the harmonizing of internal and external reality, if the internal models of high order function are relevant as they are for motor control, and cerebellum is involved in all these high order behavior dysfunctions, then we have a new way to think about mental health. And it is conceivable that the dysmetria of thought theory may underlie, at least in part, much of what we think of as contemporary neuropsychiatry. There is much to read about this. The syndrome has been replicated multiple times. Here's the work from the Italian group who've done beautiful work on the cognitive profile. Marco Molinari talks about sequencing as an important cerebellar function, part of the transform, the role of cerebellum in emotional processing and social cognition. The task force discussed this as a meta-analysis. There's a consensus paper on emotion and on social cognition. I reviewed many of the recent papers on this just last year. And the postoperative pediatric cerebellar syndrome is key also in thinking about the developmental cerebellar cognitive affective syndrome, not just in the children with the early childhood hemorrhages and trauma, but the children who had tumors taken out and then are left with the developmental CCAS as they evolve through the years. It goes into other diseases. There's a network selective vulnerability of the cerebellum to major diseases, Alzheimer's and FTD. And the location of the anatomy of the morphometric change in the cerebellum depends on where the lesions are in the cerebral hemisphere. CREST 1 and CREST 2 in Alzheimer's disease, and Level 6 in behavioral variant frontotemporal dementia. So maybe the cerebellum is not really a bystander in Alzheimer's disease, and maybe it has a role to play in these major disorders. The role of cerebellum in cognition is expanding. There's 92,000 papers in PubMed going back to 1804 on cerebellum. 8% of these from the last 30 years are on cognition. And this is increasing. So the role here for cerebellum and cognition is a big deal. Here's a summary of my talk in one slide. So the what of the syndrome is a triad of clinical ataxiology: the cerebellar motor syndrome, the vestibular syndrome and autonomic features, and the cerebellar cognitive affective syndrome. Why is this? Because of dysmetria of thought. The cerebellum does for cognition and emotion what it does for motor control. How is this possible? It's possible by virtue of the universal cerebellar transform enabled by the repeating architecture of the corticonuclear microcomplexes and the functional topography of the sensory-motor, vestibular, cognitive, and limbic circuits with extracerebellar structures. And the importance of this, the so what, therefore, is the need to know imperative to acknowledge, recognize, and treat the cognitive affective syndrome in patients with cerebellar injury, and then to think about cerebellar interventions in neuropsychiatric disorders as we understand the reach of the cerebellum in beyond cerebellar motor function. So he has this lovely view of a cerebellar folia, and the bottom line is that little brain, big deal. So with that, I'll end and pay tribute to my many collaborators and teachers through the years. I've mentioned them as we've gone through here, and to also pay attention and honor the funding sources that have helped us get to this point through the years. Once again, thank you for the great honor of the invitation to present the Fernando Rossi Lecture. And I am happy to use this opportunity to draw attention to and mark respect for my many colleagues in the field, both in the society and outside, who are looking at the cognitive neuroscience of cerebellum, and to also recognize that the work that's been going on for many years in trying to understand the details of cerebellar anatomy, physiology, neurochemistry, and genetics has relevance not only for the cerebellar motor syndrome, but for all these other aspects of cerebellar function, from movement to cognition and emotional processing. So I'll end there and thank you for your time.
Thank you for your excellent lecture, Jeremy. Now it's open for discussion. Please type the questions to the Q&A system. I would remind that speakers and session chairs cannot use the Q&A system. So if you have questions, activate your camera and microphone and ask directly. In the chat, what is another possibility, we have a question by Haider Jinah, and he wrote that he has seen the cerebellar affective syndrome after cerebral injury in children. Does it also occur with cerebral injury in adulthood?
So yes, the syndrome was described in adults. So that's where we originally came up with the whole idea of the cognitive affective syndrome in focal lesions in adults and in the degenerations in adults. And now we know from work coming out of the Cerebellar Research Consortium for the study of the cerebellar ataxias that patients with the cerebellar degenerations describe all of these difficulties as well. So this is certainly in adults. And in children, it's profound. So as I've showed in the youngster who I showed at the outset, the children with the cerebellar hemorrhages, the agenesis cases, the early childhood injuries, these children are left with the whole range of phenomena that are part and parcel of cerebellar cognitive affective syndrome. And our first demonstration of this was actually in 2000 in the children who had tumors taken out. And then shortly after that, Riva and Georgi also showed in 2000 that the children who they studied had a variety of deficits, including autism spectrum behavior. So this is very much an issue for both adults and children throughout the spectrum. And the notion of a developmental cognitive affective syndrome from early childhood brain malformation is certainly key. Something is as common as Joubert syndrome, not that common, but as a well-known phenomenon, has its predominant features as the neuropsychiatric rather than motor. So the answer is very much so, yeah.
Thank you. And in the Q&A system, we have another question here by Mario Manto. In the motor domain, dysmetria and tremor are two major features. It is now established that dysmetria applies to both motor, cognitive, and affective domain. Is there evidence of a shaking presentation, similar somewhat to tremor, in the field of cognitive, affective, or social operations?
Yeah, thank you, Mari. That's a wonderful question. I have to think about that because the tremor has such a short temporal characteristic to it that you can think of the marked fluctuation of behavior in the pediatric postoperative mutism or cognitive affective syndrome, where you can have irritability on the one hand and then fluctuating rapidly with happy, giggly, and then unconsolable, irritable on the other. A rapid cycling of behavior within a matter of the same examination. I don't know of a shorter time span. That will be interesting to try and figure out. The time scale for movement analysis on the one hand and behavior on the other is somewhat different. So the closest I can concertina that time frame down would be almost like bipolar disorder manifesting in the same interview or the same examination period of time. But it's a wonderful question. I'll think about that some more.
Jeremy, I have a question. First of all, congratulations for this wonderful talk. I have two small questions. Do you know how CCS evolved with aging? First question of people.
Yes.
And the second is, what is known about potential treatment of CCS?
Right. So there are a couple of things. There is some work. This goes back a while. I think that Anna Woodruff-Pack actually may have looked at this, looking at cerebellar changes with age. And so we know about normal aging cognitive change. The extent to which that normative cognitive aging has a cerebellar signature, I think, is a very open question. We know that there are cerebellar changes with age in terms of the morphometry of cerebellum. We know about the cognitive changing with age. What we don't yet know is, and it's been an assumption so far, this is all cerebral hemisphere, is there something about a cerebellar signature for normative cognitive aging? It's a great question, and the answer is not available. And then,
I mean, if you have somebody which has already a CCS syndrome and you have the chance to follow him during aging, do you see even only clinically some changes that were remarkable or not?
So I think it depends on the nature of the initiating event. And I think you get directly into the issue of plasticity here. So I don't know the answer for sure, but here's for the patients I've followed, here's my sense of the answer to that. People who have a single injury, we showed in our initial study, will recover over a period of months. If it doesn't involve the nuclei, they do better. So the executive function problem remains troublesome, but many of the impairments will improve over time if it's a single injury. Those who have a progressive injury obviously get worse over time. So now you post-stroke essentially. Mm-hmm. So your reserve over time will probably be less robust than it would have been had you had a brain that was entirely undamaged. So I think that the answer will depend on the nature of the injury and the nature of the subsequent change. I have seen personally a patient who a very high functioning person who had a previous cerebellar stroke, came in with a metabolic encephalopathy and had a language impairment that then resolved over time. So it was a recrudescence of previous injury from a metabolic change. So I think from that perspective, cerebellar injury will obey the rules of other prior injuries in recrudescence. And then you had another question that I liked as well. What was the second part?
The question was about what are potential treatment of CCS?
So at the moment, what we're doing for the treatment is we're repurposing treatments for other disorders for cerebellar diseases. So for example, we'll treat depression with the SSRIs, or if there's psychiatric phenomena, we may use the atypical antipsychotics. But I think that and then there is behavioral therapies for executive control. I think what's going to be really interesting down the road is, as we talk about cerebellar TMS and TDCS, these are going to be important aspects of the approach. I didn't tell you about a patient who we still have to write up, of a young girl who had cerebellar hemorrhage devastated 3 years later, cognitively, emotionally, motorically not so bad. And we gave her TMS twice a day for a week, and she got better. So I think there's something about the brain stimulation and its ability to evoke improvements in the cerebrocerebellar interactions that is going to make an impact on the functions arising from cerebellar disease. Hasn't yet been that successful for ataxia, but I think it's going to be helpful in the role of cerebrocerebellar interactions for neuropsychiatry. The other piece of this in terms of therapy is, and I ended off with this, I think that the approach through the cerebellum, through brain modulation of psychiatric disease is just waiting to happen. That if you upregulate the brain circuits through the widespread manipulation of the cerebrocerebellar interactions, we have a new opportunity to increase the modulation of those circuits and therefore improve behavior. So I think that's where this is going.
Thank you.
Pavel Němec.
Thank you for excellent talk. My question is, there are some recent comparative studies that use measures volumes of vermal and paravermal regions and interpret it as substrate of motor functions versus volumes of lateral cerebellar hemispheres and interpret it roughly as substrate for cognitive functions. Do you think that this broad division of cerebellum is too oversimplified, or it's acceptable, or what do you think about like this broad division of medial and vermal and paravermal region and cerebellar hemispheres?
I think it's very reasonable for the stage that we're at the moment. As we get more sophisticated with our structure function correlations and depending on the different parameters of evaluation of each center, people will start to be able to look at lobule by lobule analysis. But I think it's certainly for the clinical domain, I think that's very useful. Work that just came out from Mark Helko and his group a little while ago showed that within lobule 7, you can differentiate different aspects of attentional modulation. Almost like the what and where syndrome in the cerebral hemispheres. So the cognitive neuroscience questions are becoming much more sculpted within the different lobules and folia. But I think for the clinical aspects, differentiating the vermal, predominantly limbic, from more lateral hemisphere, predominantly cognitive, is a good way to start, and that will evolve over time.
Thank you very much.
So, we are a bit late, but I think if we shorten the pause, we can realize few or very short question and answer. I saw Pascal Hilbert that appeared and Detlef Heck. So who wants to ask?
Yeah.
Can I? Okay.
Sorry.
All right. Yeah, go ahead. Go ahead.
Oh, sorry. It was about the treatments. Thank you so much for your presentation. It was wonderful. And I asked the question: if we change the frame of thought, that is, if we envisage that the cerebellum is really involved in psychological disease, what does it change from treatment point of view for these patients? And is it some interaction with classical treatments? And from a psychological treatment, what can we envisage now if the cerebellum is involved in schizophrenia or anxiety disorders or depression?
I think it's a very important question, and I have to tell you, I don't think that I don't have the answer right now, and I think that where we are at the moment is just the fact that we can ask these questions is a new day. So if we ask these questions, the end line of my paper in 1991 was, by drawing attention to these questions, we can start to address the answers, and that's what's been happening over the last few decades. So the question that you're asking is there something specific about the intervention that derives from the cerebellar involvement in these diseases? I think it's a really important question. At the moment, we are repurposing like they were doing in psychiatry many years ago. Is there a targeted approach to therapy? We're going to have to find that out. But I think that the idea that we can use cerebellum as an entry point for interventions in psychiatric disease is the opening salvo of a whole field of science of therapy for the neuropsychiatric disease. So I think that's where this is going. So I think that your question is exactly well put, and understanding what the answers could be could help change the landscape for patients with these very, very intractable diseases.
Yeah, I think the cerebellum will be a checkpoint for the treatment now.
Yeah, right.
Thank you. Detlef Heck.
Yes. All right, Jeremy, thank you very much. It was a great talk again. I wonder if you emphasize the homogeneity of the cerebellar architecture, which of course is true in a sense, but there are all these sagittal zones. I wonder what your thoughts are on whether or not they play distinct roles. Is there something you can, what do you think about the meaning or the relationship between these sagittal zones and cerebellar cognitive versus motor function?
So I think if you stay with I think that's a great question. If you stay with the reductionist view, I think the answer has to be that they do have a sculpting differential role. I think there has to be. We've known from your work and the work from Christa's user lab in terms of the different firing rates of the Bezievin positive and negative zones. All of the sculpting detail that goes on into understanding the underlying neurophysiology of cerebellar cortical microcircuits, I think is key. There will be there has to be some functional correlate of those relatively discrete but clear differences in its organization. So the big picture of the organization is as it is. The sculpting of it must have some functional relevance within the overarching framework of what the cerebellum does for modulation. So I think the answer is yes. Exactly what it's going to be, I'm excited to learn from you all about what that would be. At the level of the patient and the big lesions, we can't tell. That's going to have to be at the level of microcircuitry stimulation and the whiskers and the rat and so on to try and figure out how does that play out in the non-motor domain. Yeah. Or in the motor domain itself.
Okay, I have realized that Professor Scholek wants to ask something or react. Please, if you could open your microphone because you are a panelist. And meanwhile, there are still some questions in the Q&A. Is cerebral cognitive affective syndrome also leading to a gradual degeneration of other more cognitive areas such as the hippocampus?
So absolutely, there's some beautiful work now on hippocampal cerebellar interaction. I see Laurie Rodriguez on the line, and she's worked on this herself. So this and the group in Iowa has done similar work. So this interaction of hippocampus and cerebellum has relevance for spatial navigation, for spatial learning, perhaps for other aspects of cognitive limbic interaction. So whether it leads to degeneration in hippocampus, I'm not sure about that, but certainly that interaction of cerebellum and hippocampal formation, what we learned from Heath years ago on cerebellar interactions with limbic circuitry in the septal nuclei, what we know now about the cerebellar interaction with the brainstem dopaminergic systems, all of those limbic circuitries or paralimbic circuitries in their interaction with cerebellum are now ripe for the picking in terms of understanding the neurobiological basis of these neuropsychiatric phenomena. So I think it's a key question, and there is something about that interaction. I'm not sure it leads to degeneration, but certainly that it taps into that connectivity that will change behavior by virtue of the cerebellar injury itself.
Thank you. And the very last question I would like to ask for a very short answer. The question by Maria Cvetanovic, great talk, Dr. Schmachmann. Is there any evidence of changes in the activity of deep cerebral nuclei with cerebral cortical injuries? What are your thoughts whether deep cerebral nuclei can compensate for cortical injuries.
This gets to all the beautiful work going back a long time on the cortical nuclear microcomplex from Masao Ito on. And there's no question that if you damage the deep nuclei, you have a greater injury because you've affected the outflow back into the hemispheres. Is there compensatory capacity? I think the answer is yes. We know this from the work of Dagmar Timmerman and her group in the motor phenomena, that if you have the deep nuclei involved, the motor syndrome doesn't recover as well. So that the degree of involvement of the cortex versus the nuclei really matters. And even go back to Bowman and Kemper in the autism literature, they thought that there's something about the nuclei and the cortex that is important for the differential presentation of the autism at different ages. So the nuclei are absolutely key in this consideration, whether they can compensate or not, probably, but certainly if you damage the nuclei, you have an even exacerbated syndrome because you've now affected the influence back up to the cerebral hemispheres from the limbic and cognitively relevant regions of the cerebellar cortex.
Thank you very much. Jeremy, thank you for excellent lecture, for the answers. And I would like to thank to all people who posted their questions. And now let's have 5-minute break only before the next session. Thank you.
Thank you so much.
Morning and good afternoon to everybody. I think we are more or less in time to start for the first session of this first remote international symposium of the society. The start has been really great. I must thank Jeremy for the marvelous presentation. And now we move to this pathophysiology and therapy of cerebellar disorder. This is the first session. And I'm very happy to introduce Rachel Sherard that will lead us to the capacity of repair using low intensity magnetic stimulation. As Jeremy was saying, neuromodulation, cerebellar neuromodulation is one of the main avenues toward for the future of cerebellar therapy and the influence of cerebellar as a window for treatment of different disease. So let me introduce Rachel Sherard and remind you that you can make question and answer writing throughout the presentation, and I will try to put your question to the speaker. Thank you. Yes.
Okay, thank you. Thank you for that, Marco. That was extremely kind. Like you say, Jeremy has raised some huge questions about what we can do to modulate the cerebellum in terms of treating the large array of diseases. I don't pretend to have any type of an answer, but I'm very grateful to the organizers for giving me this chance to show a bit of our work on low intensity magnetic stimulation on the cerebellum and what it does. So, as everybody knows, the brain requires, in order to function, requires extremely precise connectivity. And this needs to be interregional, as we can see here, but also needs to be very specific at a cellular and subcellular level. So therefore, if there is a lesion, we need to be able to recreate these connections with a similar degree of cellular and subcellular specificity. This is particularly true for the cerebellum because, as Jeremy has just so brilliantly described, and as Mary Hatton has done as a nice shimmer here, the cerebellum has extensive connections throughout the whole of the forebrain and therefore has a large number of functional problems when it is injured. Our interest is in magnetic stimulation and what it does to the cerebellum. We started off with just wanting to see what it does to a normal cerebellum, because we don't want to mess up a system that is already working. But we've also been interested in olivocerebellar reinnervation, and most importantly, how does it work? So here, if you have a large coil stimulating the cerebellum of a human, we have a problem when you're looking at mechanisms. Because if you want to work with a mouse isn't the same size as a human. And so therefore, our first task was to create a coil to stimulate a mouse cerebellum, and it was therefore an appropriate size. We did this, we've targeted it towards the cerebellum and measured the intensity of the field in different regions of the head from that coil. What we have decided to do was we took normal Joe Public average adult mice. We stimulated them for 10 minutes a day for only 2 weeks. And then we looked at their behavior and their cerebellar morphology, and particularly the Purkinje cells. What we found was their basic motor function, and including their coordination on a rotor rod, was completely unchanged. So we pushed a bit harder and went to spatial learning and memory, which requires a cerebellar contribution. We did 6 trials per day for 5 days, in which the mouse has to use external cues to find a submerged platform in this water maze in order to escape. What we found is, as expected, the mice searched fairly randomly to begin with, and then learned how to find the platform. Both ordinary mice and stimulated mice learned the task very well. And there was indeed a suggestion that the stimulation improved the speed of their learning. But we needed to test this with a little bit more detail. We checked their memory. And to do that, we used a probe test 24 hours after the last learning phase. And in this probe, we removed the platform from where it was. And if the mouse knows where it's going, it usually swims there and swims around searching for the platform that we've unkindly removed. So if you look at a heat map of the pool, their presence is concentrated in the region of the platform. So what did our stimulation do? Well, both sham and stimulated mice spent a greater time than chance in the quadrant where the platform had been. So it implies they knew where it was. But also the latency to get to the platform and the number of times they crossed over the platform were improved in the animals that had been stimulated in compared to those with sham. In this situation, so therefore we could say that our magnetic stimulation at low intensity facilitated at least this particular cerebellar cognitive function. We then wanted to see what the underlying basis of this was. Now, there could be millions of tests to look at, but we decided to start with the basics and look at Purkinje cell morphology. In this situation, we filled Purkinje cells and measured the area occupied by their dendritic tree, and found that it was significantly increased in the animals that had been stimulated. When we looked at the spines on these dendrites, they too, their density was increased. So therefore, with a greater density of spines on a bigger dendritic tree, this suggests that our low intensity stimulation modifies the connectivity of Purkinje cells because there are more available sites to receive input. So we know that there is some effect on the normal cerebellum, but of course the key is, can we use it to treat problems in the cerebellum? Our particular area of interest is the olivocerebellar circuit, because climbing fibers are fairly fundamental for cerebellar function. This pathway goes from the inferior olive in the brainstem, terminating on Purkinje cells organized in parasagittal bands in the contralateral cortex. We know a great deal about this pathway, both in development and the fact that post-lesion it is capable of repairing itself. This particular reformation of the pathway is if the peduncle is cut on one side and growth factors, and I give BDNF as an example, are injected into the denervated hemicerebellum, the remaining pathway can develop collaterals which cross the midline and reinnervate the Purkinje cells. This reinnervation is useful in the sense that it terminates correctly on Purkinje cells, its synaptic function is normal, and it gives some recovery of function. This is the path taken in a Morris water maze to get to the platform, which is not completely better, but it is better than those who do not have any reinnervation. Now that is with a growth factor. What we want to see now is, can we use our magnetic stimulation to reproduce this effect? So again, we took perfectly ordinary adult mice, cut their peduncle and stimulated them post-surgery for 2 weeks, targeting the cerebellum and mapping the intensity of the magnetic field therein. What we found was that in the molecular layer of the left hemicerebellum, there was V-glute terminals, which are consistent with climbing fibers. And when we mapped their distribution, we find that although they were mainly in the vermis, some of them did extend out into the cerebellar hemisphere. And also when we looked at the amount of reinnervation per lobule, it seemed to be roughly parasagittally organized. So this is an optimistic start. But if we want to start thinking about how does magnetic stimulation do this, which of course is always the big question, we decided to leave in vivo mice because this is a really difficult surgery to do. And we went to the in vitro olivocerebellar explant, which was initially created by Alan Shedd et al., where the hindbrain of a mouse embryo, which includes immature cerebellar components and the inferior olive, can be taken, flattened, and cultured on a membrane for a good 6 to 7 weeks. Mathieu Letellier, who was a previous student of the lab, showed that these explants develop effectively normally, developing granule cells and parallel fibers and climbing fibers, which terminate on different component parts of the Purkinje cell dendritic tree. We have also shown that we use this to reproduce our in vivo lesion by putting a hemicerebellum, removing it from one explant and putting it next to another one, and following the process of reinnervation. So the question is, let's do it with magnetic stimulation and see what happens. This was another problem because human coils will not tell us what is going on inside a culture dish, and equally our small mouse coil will not give a consistent homogeneous magnetic field to the tissue that we're trying to stimulate. So we had to generate a new one in which the explant could sit in the middle so that it received a homogeneous magnetic field, shown here by mathematical modeling, and that our areas of interest in the cerebellum and the olive received similar electric fields. We used this with a range of stimulation frequencies and looked at olivocerebellar reinnervation, seen here as Purkinje cells co-localized with V-glute2 labeling. We also analyzed close to the host tissue and further away as proximal and distal zones. The message of this graph is that with 6 different parameters tested, 2 were highly successful at inducing reinnervation, 1 had a marginal effect, and 3 others did nothing at all. So this shows that you have to find the right protocol of stimulation in order to have a positive effect. The other thing that is important about this slide is that when you look at the number of pulses delivered compared with the reinnervation, there is absolutely no correlation at all. And this is slightly contrary to the perceived wisdom of human stimulation, where it is thought that the number of pulses is important. So how did this happen? First of all, we looked at what neurons are, respond to our stimulation. There are a lot of different types in the cerebellum. And initially, when we made CFOS counts, it didn't make sense. This stimulation didn't induce reinnervation and this one did. So is this any relevance at all? But when we looked at the appropriate populations of cells, here Purkinje cells, which are positive for CFOS or even parvalbumin interneurons, we find that those patterns of stimulation which induced reinnervation induced a greater percentage of cells that were positive to CFOS. So it seems that Purkinje cells are activated, and this seems pertinent to the reinnervation. So to find out what was happening, we took our tissue that had been stimulated. We took it after 3 days of stimulation because we wanted to see what was going on during the process of reinnervation. And we did an array, a qPCR array, looking particularly at neurotrophin signaling. And these two parameters induced reinnervation, and they strongly upregulated genes, whereas one that did not have that effect. But what we also found interesting was that a significant proportion of these upregulated genes were enriched with geo functions of axonal growth, reinnervation, synaptogenesis. So this would imply that our low intensity stimulation increases growth promoting genes in association with reinnervation. So, okay, that's really nice and very encouraging. The but is there is a huge black box between stimulation, magnetic stimulation on one side and gene expression on the other. So how does these two get together? This requires that we know a little bit about the cellular mechanisms of magnetism. And this is where it gets a little complicated and controversial because people are not always in agreement. It is known that a magnetic field will induce an electric field and therefore an electric current in appropriate tissue like the brain. And it is thought that this electric current can open ionic channels and depolarize neurons and make them active. And indeed, this is one of the mechanisms that is thought to underlie, in particular, RTMS, but possibly also TCDS applied to the brain. There is the presence of magnetite crystals that can align with a magnetic field and therefore mechanically distort channels and allow them to depolarize cells. But in fact, these have only been proven in certain cyanobacteria, and there is no real evidence that they exist in birds or mammals. They are not what is present in pigeons' beaks, despite what has been said. There is, in fact, a third component, and this component is the one that has been up to now really very controversial. And this is a protein called cryptochrome. It is a flavoprotein that is known to be able to respond to magnetic fields because it sits in an oscillation of electron transfer between an oxidized and a reduced state. And this electron transfer is associated with a free radical. In the return to the oxidized state, there is this electron transfer results in the production of reactive oxygen species. So which of these might be involved in our stimulation that results in differences in gene expression? First thing we did is we thought electromagnetic because that's what is the norm for magnetic stimulation. So we looked at intracellular calcium and found that at the end of our 10 minutes of stimulation, it was significantly increased after different types of stimulation. When we did the appropriate pharmacology, showing whether it was an influx from the external milieu or whether it was released from intracellular stores, we found that it was in fact released from intracellular stores. The pertinence of this is that this is entirely consistent with a very low magnetic field inducing a low electric field that is not of a degree of magnitude capable of inducing direct neuronal firing. So that seems to be appropriate and consistent. If we're considering intracellular release of calcium, This is through IP3 and ryanodine receptors on mitochondria and endoplasmic reticulum, which also play a role in cellular redox modulation. So what? Well, this cellular magnetoreceptor, cryptochrome, produces ROS in the return phase of its metabolism. And here it is in basically its non-activated state, and it is oscillating all the time between its oxidized and reduced format. And in this electron transfer circuit, we produce a small amount of reactive oxygen species. But Cry blocks the transcription factors BMAL and clock. When it is activated by a magnetic field, the formation of this protein changes, which releases the block on the gene expression. And indeed, we found changes to gene expression, but also the cycling of the free radical and the electron is greatly increased. So there is an increase in ROS. Could this be the mechanism that underlies what our magnetic stimulation does to the cerebellum? Well, what we did is we started off looking at something very simple, which is in fact an embryonic fibroblast, because we could get them without the Cry magnetoreceptor. And what we found in those that were normal, there was an upregulation of ROS. This is a fluorescent label in these cells. Whereas in those that are knocked out for cryptochrome, there is no such increase in reactive oxygen species. So it is clear that the magnetic field requires Cry to produce this ROS. We then went back and looked at our regulated genes to see whether they had anything to do with Cry. And we looked at potential response elements in their upstream promoter regions to see whether they could be a Cry target. This would have to go indirectly via BMAL and clock. And 9 of our 22 genes contained appropriate response elements. It doesn't mean that they are targets of Cry, but it reinforces the story that in actual fact, these might well be involved. So we then went the whole hog and we obtained mice that would knock out for both types of Cry. And we found that even when we stimulated them, we could not induce Purkinje cell reinnervation, in contrast to this stimulation protocol that does normally work very effectively. But this was not because they couldn't grow the axons, because when we treated them with BDNF, lo and behold, we got, as it was expected, a nice amount of reinnervation. So this suggests that magnetic stimulation to our explant cerebellum might actually be inducing reinnervation by its magnetic field rather than the induction of an electric current. So what do we have? We have a study of low intensity magnetic stimulation to the cerebellum. We know that it can make minor modulation to a normal structure, but it is capable of inducing axonal growth and reinnervation to repair part of the cerebellar circuit. This may well be by calcium flux within neurons, particularly Purkinje cells, and we know that the repair and changes to gene expression are frequency specific, and this is the key. We need to know what we're giving to our cerebellum if we want to treat it, and that it indeed requires the presence of a magnetoreceptor in the cells. So this leaves 2 questions. One is, when we're stimulating the cerebellum, do we need to use high intensity, or could something a little bit more user-friendly at low intensity that could perhaps be taken home and used as domiciliary treatment, will that do? And the other thing is how can we identify ways of finding appropriate parameters to stimulate, given that we know that it can be effective. Thank you for listening. I want to thank all the people who have actually done the work, like my PhD students and postdocs, and my collaborators in Western Australia with whom I have worked closely. So thank you if you have been listening.
Thank you very much, Rachel. It's been very interesting presentation, and I think it's opening up exactly some ideas quite important to understanding what's happening in this quite black box that is neuromodulation, in which we are doing a lot of things to the patients, but we don't know exactly what is happening. I would like to open the question and answer time with a direct question. Your data are all focused on the cerebellum. How much are these data can you apply also to other neurons, to other areas, in generally to neuromodulation as a whole?
I presented this as cerebellum because it's a cerebellar conference. We have also looked at stimulating the hippocampus in an Alzheimer's disease model and found that there are positive effects there, including on cognitive function. And interestingly, the most effective stimulation patterns are not the same as they are in the cerebellum. We have also looked in the cerebral cortex to see what some of the stimulation can do to neuronal activity. So it's not just in the cerebellum. I've presented it as a one structure simply because of the nature of the conference.
Yes, so is area specific or not? Are the changes in the cortex similar or equal to those you see in the Purkinje cells, or are different? Let's say the frequency and the effect on changes in the calcium activity is different?
Some of this is well, this is all ongoing. So we haven't looked at the calcium in different cell types. We do know that cortical pyramidal cells do have their activity changed. We're in the process of writing the paper for that. We also know that in the hippocampus, for example, in an Alzheimer's mouse model, we can improve spine numbers and spine density, but interestingly, that's with 10 Hertz rather than with the ITBS and the BHFS that we find in the cerebellum. So different tissues respond to different stimulation parameters.
Thank you. We have also a question from Owen Chow, who is asking if the TMS stimulation is on or off during, pre, or after the task that you are doing with the rats on the special maze, I think.
It was before. We used it for 2 weeks to hopefully induce re-innovation, and then did the behavior afterwards.
Okay, we have another question from Maria Chetnovich, who is asking if, first giving you compliments about the Greek talk, and now is how you examine any of glial cells, astrocytes, microglia, oligos, and so on.
I haven't personally, but my colleague in Australia, Jennifer Roger, and her lab have looked at glial cells, and they have found that, for example, around a cortical lesion, there is a reduction of gliosis and GFAP following different stimulation protocols. I'm not sure that they've looked at microglia. The other interesting one would be to see whether we can induce myelination to think about treatments for things like multiple sclerosis.
We have another last question from Mario Manto, who is asking if the cellular response to simulation is associated with the morphology of the cell, especially the size of the soma.
I don't know. In fairness, we've only looked at large projection neurons. So in the cerebellum, it's Purkinje cells, and in the hippocampus, it's the larger pyramidal neurons. And indeed, their response, we have seen spine changes in both of them and of the dendritic trees. That is, it's difficult to say. That is, on an electrical induction point of view, that is highly probable, because bigger cells are going to be more susceptible and more responsive with respect to the stimulation. Given that we are thinking that some of this actually might be running through cry, it's more down to the levels of expression of cry. But that is something that perhaps needs to be looked at, looking at granule cells, for example.
We have another question from Dr. Eck. Can you?
Yeah, hi, thank you. That was a fascinating research. I have a question about when you stimulate the hippocampus. Have you looked at changes in the cerebellum after that stimulation?
No, we haven't, and we should have done. It was a bit stupid. In fact, what we're doing at the moment is we are doing some cerebellar stimulation and looking at changes in areas of the cerebral cortex, and vice versa, stimulating the cerebral cortex to look and see what happens back in the cerebellum. But that's fairly new work with a PhD student who's only in her first year, so there's a lot to go on there. But yes, I suspect the long distance connectivity turns out to be very important.
Thank you.
Tim, I see your camera on. Are you going to ask a question or?
Yes.
Okay.
Rachel, that was fascinating. I was particularly intrigued by the changes in the Purkinje cell, both the increase in the tree and the dendritic spines. Do you know, is it transient? Is it permanent? How long do those changes last?
That's a good question. For the spine changes, now we were looking at those mice. They weren't killed immediately. We looked at those changes at least 2 weeks after the end of stimulation. So that shows that they're not transient in the sense of minutes, but whether that means they're permanent is a moot point. The re-innovation by climbing fibers, that's permanent. That stays there.
Okay, thank you very much, Rachel.
Thank you.
We have to move on. Thank you very much again. And now I'm going to introduce Lorenzo Magrassi from the University of Pavia, and he will talk us about the capacity of xenograph to survive in the cerebellum. Another type of therapy, and we are very keen to hear what he's going to tell us.
Okay. Thank you very much for the invitation, and it's a very nice meeting. And I want to share with you my experience in xenograft, in studying xenograft in the cerebellar system, an experience that started with Ferdinando Rossi many years ago. And in fact, while it became available, many protocols and many labs that were using human-induced pluripotent stem cells differentiated towards neurons of the cerebellum, we thought that maybe that could be a good way to obtain a model to study the differentiative potential of this cell. And all this is based on a fact that is known from many years, at least should be basic stuff in immunology, that if you graft some foreign, some xeno cell before the complete development of the immune system, you should be able to induce immune tolerance and in such a way obtain that the cells that comes from a different species will integrate and remain in the animal for a long time. And indeed, we were able to perform this kind of experiment by using intraembryonic, intrafetal injection of mouse cells into a rat. And the mouse Purkinje progenitor will integrate and survive for the entire life of the rat. In doing so, sometimes you could even show that they can survive even more than 2 times their supposed maximum lifespan if they were remaining in the species of origin. So we thought if we start with human induced pluripotent stem cells that were partially differentiated in vitro following any of the multiple protocols that have been already published, we should have been able to obtain the same, result. And indeed, we started with embryoid bodies. The embryoid bodies were dissociated and cultivated in vitro in the presence of growth factors, especially FGF2, and to commit their differentiation towards the cerebellar root. And then before they became totally they became post-mitotic, we dissociated them and injected into the cerebellum in the embryo. Usually we target, we did that both in rats and mouse, and we usually target the 4th ventricle. We also did some experiment in neonatal mice because this tolerance induction protocol should work also injecting the cells before the 5th postnatal day, at least in mice. And very, as we expected, we got a nice taking of the cells that when we looked one day after the injection, when the fetus was still in the womb of the mother, we could see cells integrated in the ventricular proliferating zone. We could see cells integrated and connected to the choroid plexus, that is a source of growth factors and other factors that are very important for the differentiation of the cerebellum, and other that were already in the developing cerebellum. And so we went to check for these cells, their differentiation, 30 days after the animals were born, and we found, in this case in a rat, that the graft took is positioned in the white matter of the cerebellum. And when we studied the differentiation of this kind of graph, we found, as expected, that they were coming from human cells using this human nuclear antigen, an antibody against a human nuclear antigen that is not present in rat and mice. And some of these cells were also expressing very early markers of differentiation. We shouldn't remember, and then I will stress this after in the next few slides, that the timing of differentiation of the human cerebellum is much longer than the timing of the differentiation in the mouse and rats. And so these cells, even though were already implanted from more than 30 days in the rodent cerebellum, were still expressing, were not expressing neuron markers, and we're still expressing beta tubulin, so they were very, very immature. The same thing we obtained when we transplanted the cells in the developing cerebellum of the mouse. Why we tried both rats and mouse? Well, because as many of you are aware of, other people that have developed a similar paradigm trying to induce immune tolerance to allow injection of human cells into the rodent brain at later stages, that the stage that we are working with, discovered that mouse is not easy to induce a good immune tolerance for these cells, and rat is better. So we tried on both species without any difference. But after 1 month, when we, as we programmed before, was planned before, we tried to sacrifice, we sacrificed mice and rats that received the transplant before the complete development or even the initial development of the immune system. We looked after the first 30 or 40 days, we didn't find any transplant. We found after serial sectioning of the brain of long-term surviving mouse, some cells that were EGFP positive, but these cells were probably cells of the host that engulfed the human EGFP containing cells, because if you look at the chromatin structure of their nucleus, like in this case it's very clear, it shows a clumped chromatin that is typical of the mouse, very different from the chromatin that we see in the human. So we did this, we repeated this several times at different length, and it was very clear that everything went fine for the first 30, 40 days. But after that period, both in mice and rats, all the cells disappeared, and only remnants of the protein, of the human protein, were still there. So we thought, well, it may be due to the fact that the cerebellar development takes approximately 35 days in mice and 1,040 days in humans to complete. And if you make a ratio, a very gross ratio between the developmental time of the two different types of cerebellum in the two species, it comes around 29.7. And it's quite interesting that if you compare the different weight of the two cerebella, well, initially, of course, it comes out completely out of range, about 3 orders of magnitude of difference. But as you remember, the weight is certainly has to be considered a three-dimensional mode of comparison. So the best thing to do is obtaining the third, to make a third root, and then you still have a linear 13.48. That's very similar to the average difference between the ratio between the developmental time. And why that? Well, we thought that if you have cells that are still if the human cells still follow their the developmental program that they have in the human in vivo, well, it is well possible that we induce tolerance only for the antigens that were present during the development of the immune system. And when these cells start to develop more advanced antigens and features, then some of these antigens will not be recognized by the host immune system as a self, because they were not present initially when we transplanted them, and so they are rejected. And to be sure that this was due to some kind of rejection, we, of course, try to transplant the same cells into a non-skid mouse model, as many others have done, and indeed we obtained a very good transplant taking, and this transplant were also very slow to differentiate. Remember, these cells, these iPS cells, were only induced in vitro towards the cerebellar differentiation but they were not terminally differentiated. We were not transplanting cells that were already showing, as an example, Purkinje cell characteristics in vitro before the transplantation. So these cells were already committed toward the cerebellar pathway, but development, but they were not definitely differentiated as a neuronal projection of typical cerebellar cells. The problem with the non-skid mouse, is, as many people knows, that these non-skid mouse usually don't last more than 6-8 months because they have many problems, and so you cannot study the entire development because you need at least a couple of years if the rate of development of the cells is the same in human. So you need a very long survival time. And so non-skid mice, although a very good model, are not the optimal model. And furthermore, they lack completely any immune activity against the transplanted cells. So you cannot study the interaction between the two different environments. So there is another point that I would like to point out to you, and this other point is, as I said, the cells that we were transplanting were not completely were specified for cerebellum, but collected while they were still proliferating, and we did not try to transplant cells that were more differentiated. But if you look at the published data about the timing of that you need to obtain Purkinje-like cells in vitro, starting from embryonic bodies derived from human iPS, and you compare it with cells that have been treated always in vitro with very similar factors and techniques, you will notice that there is some difference between the timing, the time that you need to obtain well-differentiated cal-binding positive Purkinje cells in mice and humans. As you see, on average, taken from the published literature, it takes about 38 days for mice and 67 days for humans, with a large standard deviation, as you can see. But when you do statistics on this data, you don't reach any significant, any statistical significance between the difference between the two. So, although, and furthermore, as you can see, the difference is certainly not in the order of 1 order or more, like the actual differentiation of the same group of cells in the mouse. So, since we thought that the problem with this kind of experiment was mostly linked to the fact that when we transplanted them initially, they were still very undifferentiated and did not present all the antigens present in the normal cerebellum. One thing that was quite obvious to try was to try to transplant before the complete development of the immune system of the host, more differentiated cells. And we saw that there was a slight increase in the survival. So on average, the transplant of cells that were transplanted at this stage, collected for transplantation at this stage, were rejected 30 days after birth of the animal. While when we took cells that were transplanted in a more differentiated status that were post-mitotic, the rejection came 45 days after. Why such a small difference between the two conditions? Well, one possibility, and this possibility that we favor right now, is that although differentiation, it is possible to obtain some Purkinje cells in vitro, most of the cells are still very undifferentiated. And so, since we did not enrich for differentiated cells before transplantation, because doing that will reduce greatly the amount of surviving cells, we would have probably transplanted a large number of cells that were still, although post-mitotic, too undifferentiated to induce a complete range of immune tolerance. And so we thought to try a different thing. And this time, instead of transplanting more differentiated cells, we transplanted the same group of cells, and together with protein extract from adult cerebellum. And when we did that, then we obtained much longer, although in only a few animals, we obtained a much longer survival of the graft that integrated and followed the normal differentiation pathway. But as I said, we performed only a few experiments with this method, and so the maximum survival time is still too low to obtain a complete differentiation according to the developmental time of human. So what are the conclusions of our experiment? First of all, one conclusion that is very obvious is that the timing of differentiation of the cerebellar neurons and glia is cell autonomous. But this is true when you do the experiment in vivo. If you do the experiment in vitro, you can obtain a sort of acceleration that is probably only partially physiologic of the cells, of the differentiation, and you can obtain, after a very shortened time, cells that are that show antigens that are typical of more differentiated and more advanced stage of cerebellar differentiation. And the second, that is something that we are investigating right now, is to try to see what are indeed the antigens that are rejected, that are at the basis of the rejection. They certainly are not the antigens linked to a general human cells, because they were already present at the time of the injection of the transplantation of these cells. And so this could be a relatively good model of induction to study the loss of immune tolerance that, of course, is at the basis of many of the disease that are affecting the, for example, paraneoplastic syndrome, that are affecting the cerebellum, that appears to be, compared to other regions of the brain, more susceptible to autoimmune attack. Thank you very much, and I'm ready to take a question about this. I want to ask, just a second. I want to thank, of course, my co-authors that are Giulia Nato, Domenico Delia, and Annalisa Buffo, that is also a member of this society.
Thank you very much for your presentation. It's been very interesting to look at the development in the cerebellum. As before, I would like to break the ice for questions. Can you tell us how do you see this transplant in the future? There are many decades that we are looking at brain transplant in different ways, and a useful tool to study development. How do you see this as a possible therapeutic target? And what do you think can be the main problem in bridging the gap between the lab and the clinic?
Well, I think that there were many problems at the beginning, and I think that right now it looks that the source of the cells to transplant, especially with the IPS technique, this seems to be solved. There are still immunological problems because IPS cells, has been shown several times, can accumulate mutation, not so much somatic mutation, but mutation in the mitochondrial DNA, and so induce, although obtained by the same individuals or individuals that are immunologically compatible, you can still have immune response, an induction of immune response. What is far less developed and almost completely unsolved is the fact that there is truly a big difference between the volume that you need to treat in a human compared to the volume that you have to treat in a mouse or in a rat or even in a monkey. And so this difference in the size of the circuits that you want to replace or treat is still a big problem, especially because you need too many trays to uniformly distribute the cells. But of course, things can get better as they did for the source of the cell.
Thank you. Is from the panelists, because I don't see all panels in my screen, so I'm not sure if there are people that are raising hands out of the list of the panelists that are on my screen. I don't see anyone changing, and also question and answers. So thank you very much, Dr. Magrassi. It's been a nice presentation. And now we move to Francesca Malteca, just a few kilometers from Pavia. Now we are in Milano, in San Raffaele Hospital. And Francesca Malteca will talk about the mitochondria calcium axis as a therapeutic target in cerebellar ataxias. And we are moving from rewiring circuits of the cerebellum to more clinical aspects of therapy, mainly the degenerative ataxia. Please, Dr. Malteca.
Okay. So good evening to everyone, and thanks for this invitation. So today the focus of my talk will be the connection between mitochondrial dysfunction and the regulation of calcium homeostasis in the pathogenesis of cerebellar ataxias. So of course, this audience doesn't need an introduction about inherited cerebellar ataxias. This is just to point out the fact that these diseases are highly genetically heterogeneous. Indeed, we have 48 loci for the dominant forms that are also called the spinocerebellar ataxia or SCAs, that include the polyglutamine ataxias, and 98 loci for the autosomal recessive ataxia, which are also called SRCAs, and 20 loci for the X-linked. And I want to point out also that this genetic pleiotropism is further complicated by the fact that many dominant forms result in more severe recessive syndromes in case of the allelic mutations. So if we focus on ARCAs, which are less characterized compared to SCAs, we can say that the NGS has allowed the identification of a rapidly increasing number of ARCA genes, as you can see here. So given the extreme genetic heterogeneity diseases and the fact that they are very rare, the effort of researchers is now to try to identify common pathogenetic mechanism and cellular pathways with the aim to develop therapeutic approaches that can target pathways that are shared among many ARCAs. So if we cluster ARCAs based on the molecular mechanisms, we can identify three main themes. So one is mitochondrial dysfunction, then we have alteration in DNA repair and alteration in complex lipid metabolism. But it's interesting to notice that most of the ARCAs that are caused by mutations in genes involved in DNA repair have also mitochondrial dysfunction in their pathogenesis. And the best example is ATM in ataxia telangiectasia. So mitochondrial dysfunction can represent a hub for ARCA pathogenesis. And here I'm listing the major ARCAs with mitochondrial involvement. Of course, we have a free-trike ataxia, but also autosomal recessive spastic ataxia of Charlevoix-Saguenay, ARSACs, SPG7, AFG3L2, and COQ8A, and many others. So these genes will be the focus of my talk today. I want also to point out that many of these genes, mutated genes, encode for proteins that are involved in mitochondrial proteostasis. So mitochondrial proteostasis represents now a convergence pathway in ARCA. And there is also growing evidence of mitochondrial impairment also in the dominant forms, so in SCAs, for example, in SCA1, SCA2, SCA3, and SCA7. Mitochondria are multitasking organelles. They are mostly known for their role in ATP production, but they are also involved in amino acid and fatty acid metabolism and in the generation of iron sulfur clusters. But an important role of mitochondria is the one played in calcium homeostasis and signaling. Indeed, mitochondria can internalize large amount of calcium through a uniporter which is located in the inner mitochondrial membrane, which is called the MCU. And the calcium entrance inside mitochondria is important because it fuels, it regulates many calcium dehydrogenases of the TCA cycle, thus maximizing ATP production and oxidative metabolism. Another important function of mitochondria is to exert, let's say, a fine-tuning of the global calcium signals. And this thanks to the ability of mitochondria to internalize calcium in condition of intracellular calcium elevation. And in this way, mitochondria really act as calcium buffers and control intracellular calcium homeostasis. Given the fact that mitochondria are so important, they must be carefully preserved. And this is the reason why I want to introduce you to the concept of mitostasis. Mitostasis is now recognized as a specialized form of homeostasis by which the mitochondrial number and quality are maintained over time. And mitostasis consists of two levels of quality control. The first one is a molecular quality control, which is exerted by chaperones and proteases inside the organelle that preserve the mitochondrial proteome in an autonomous way. And then we have an organellar quality control, which is essentially based on mitochondrial dynamics. Indeed, mitochondria continuously fuse and divide in response to physiopathological stimuli, and they are also actively transported to subcellular site, and they are degraded via mitophagy. So the fact that so many inherited cerebellar ataxias display mitochondrial dysfunction in their pathogenetic cascade highlights the fact that mitostasis is key to Purkinje neuron health and functionality. This probably because Purkinje neurons are among the biggest neurons in the CNS, They have a high level of oxidative metabolism, and they present an extremely branched dendritic tree receiving mostly glutamatergic stimulation. So these neurons have a high demand of ATP levels and calcium buffering. So in this talk, I will provide you some examples in which impairment of any aspect of mitostasis result in the regulation of calcium homeostasis. And this could represent a convergence pathway for some inherited cerebellar ataxias. I will start with the example of the MAAA associated ataxias. MAAA proteases are high molecular weight complexes that are located in inner mitochondrial membrane, as you can see here. They can be homo-oligomers of AFG3L2 or heterooligomers of AFG3L2 plus paraplegin. MAAA proteases are important for protein quality control in the inner membrane. They also are involved in the assembly of oxphos complexes and, as we will see, regulate mitochondrial morphology. Many ataxias have been linked to mutation in MAAA proteases. Indeed, we have SPAX5, which is caused by the allelic AFG3L2 mutations. And this is a very severe syndrome with the childhood onset cerebellar ataxia, spasticity, myoclonus, epilepsy. Then we have SCA28, which is caused by heterozygous AFG3L2 mutations. And finally, SPG7, which has been for long regarded as a pure form of spastic paraplegia, but now it's pretty clear that more than 50% of patients present with the cerebellar ataxia's predominant syndrome, predominant sign, especially in case of missense mutations. So we have generated and characterized the models of AFG3L2 related ataxia. This is the knockout that, as you can see, presents with a very severe neuromuscular syndrome, and it dies very early at P14 and presents widespread alteration in CNS and PNS with the dramatic alteration in mitochondrial morphology. So this model let's say, recapitulates quite well the features of SPAX5 patients. On the other hand, we characterized the AFG3L2 haploinsufficient mice, which present gait abnormalities at 8 months of age, and they are characterized by a dark degeneration of Purkinje neurons, which is preceded by a mitochondrial dysfunction. But since these SCA28 mutations, most of them are missense mutations, we also, in collaboration with Alfredo Brusco, we characterized the AFG3L2 knock-in mouse model, which presents a phenotype which is even milder compared to the haploinsufficient one. So over the years, we have dissected the pathogenetic cascade of AFG3L2 related ataxia, and we have found that impaired MAAA function leads to the accumulation of mitochondrially encoded polypeptides. Indeed, as you can see here, this is a pulse chase in which you can see the synthesis of mitochondrial translation products, and you can clearly appreciate that in the absence of AFG3L2, many of the mitochondrial DNA encoded polypeptides are accumulating. This leads to a prototoxicity inside the mitochondrial matrix that activates the stress-sensitive proteasome E1, which in turn overcleaves OPA1, which is the master regulator of mitochondrial fusion. And indeed, the result of this cascade is an increased mitochondrial fragmentation and dysfunction. Mitochondrial fragmentation is central in these diseases. As you can see this in human primary fibroblasts, this is the fused, the interconnected network of the control cells. And you can see here that is SCA28 and even more evidently in the SPAX5 cells, the network is highly fragmented. And this is also true in the primary Purkinje cells derived from the mice. These are the alteration in mitochondrial cristae in the heterozygous Purkinje cells. And this phenotype worsens in the knockout Purkinje cells, where mitochondria are completely swollen with destroyed cristae, and they are also poorly transported to the dendritic extremities, let's say. So this fragmentation is caused by the overprocessing of OPA1, both in vitro and in vivo. Also, the accumulation of mitochondrial encoded polypeptides leads to a dramatic reduction in the assembly of respiratory chain complex 1 and 3, with a consequent reduced ATP production in vivo. And this is also confirmed ex vivo. As you can see here, knockout mitochondria are dramatically depolarized. And also, we observed a reduction in the mitochondrial calcium buffering that we measured in primary Purkinje cells using a fret-based calcium sensor targeted to the mitochondrial matrix. Indeed, as you can see here, after stimulation with KCL, the mitochondrial response in the knockout cells is strikingly reduced, and this results in a pathological increase in cytosolic calcium. So this is the mitochondrial calcium axis, let's say, that we define in EFG3L2 related ataxia. So mitochondrial fragmentation and dysfunction leads on one side to impaired mitochondrial trafficking. And this, of course, is crucial because it lowers the ATP in the periphery that is needed to fuel the calcium ATPases for calcium extrusion. And this phenotype synergizes with altered mitochondrial calcium buffering, finally leading to a deregulated calcium homeostasis and Purkinje cell degeneration. So this provided us the rationale to test in the SCA28 mice ceftriaxone. So ceftriaxone is a third generation cephalosporin, which has been proved to be neuroprotective in many models of neurodegeneration. And we took advantage of the well-known action of ceftriaxone to suppress the glutamatergic hyperactivity by increasing the levels and the activity of the astrocytic glutamate transporter GLT1, as you can see here. So we took advantage of this drug in order to reduce the glutamatergic stimulation of the Purkinje cells postsynaptically, thus reducing the calcium influx in these cells. And actually what we observed is that the ceftriaxone treatment at presymptomatic stages was able to restore the motor function in EFG3L2 heterozygous mice, and this was accompanied by a delay, let's say, in the degeneration of Purkinje cells, as you can see here. So based on this data, we obtained an orphan drug designation from EMA for the use of ceftriaxone in the treatment of cerebellar ataxias, with the aim to evaluate the efficacy of this drug in other murine models of ataxia. So, and I will give you additional examples. So the first one is RSAX, which is let's say, much more frequent compared to EFG3L2 related ataxias. We do know more than 200 mutations identified worldwide. At the moment, this is the second most common form of recessive ataxia after Friedreich's ataxia. This disease is caused by mutations in SAX gene, which is one of the largest genes in our genome, which encodes a very huge and complex protein, which is called saxin. Saxin, that is a 540 kD protein, contains an ubiquitin-like domain, 3 saxin repeating regions that share homology to HSP90, and a DNA-J domain which shares homology to HSP40. So based on this evidence, we can hypothesize that saxin could have a role in protein quality control, but this remains to be elucidated. A couple of years ago, our collaborator, Bernard Bray in Montreal, generated the SACS knockout mouse model, which nicely recapitulates features of RSACS patients displaying an early onset ataxia, which is followed by Purkinje cell loss, which is pretty confined in anterior lobes of the cerebellum. And later on, these mice present also degeneration of cortical spinal axons and loss of spinal motor neurons. This is a short movie in which you can clearly appreciate the phenotype of the SACS knockout mouse, which is smaller compared to the wild-type littermate, and it displays abnormal gait and tremor. So in a joint effort with Bernard Bray and Paul Chappell, a couple of years ago, we demonstrated that loss of saxin causes intermediate filament remodeling. Indeed, in a large panel of RSACS patient fibroblasts, we observed that vimentin cytoskeleton is dramatically altered, forming dense bundles that, let's say, accumulate close to the nucleus. And this is true in many cellular models of saxin deficiency, and this phenotype is rescued by saxin GFP overexpression. Another phenotype that is associated to loss of saxin is an alteration of the mitochondrial network morphology. Indeed, as you can see here, in the RSACS patients, the network appears hyperfused with bulb and very long mitochondria, and also the network appears to be unevenly distributed in the cytoplasm. We observed that the role of saxin in intermediate filament cytoskeleton is also conserved in neurons. As you can see here, in primary Purkinje cells, we have a clear-cut accumulation of neurofilament in the proximal dendrites of primary Purkinje cells, knockout Purkinje cells. And this is true also in vivo, as you can see here. And this is a Western blot showing the striking accumulation of non-phospho neurofilament H in the SACS knockout mice. So to gain insight into the pathogenesis of RSACS, we decided to immunoprecipitate saxin in both differentiated SHSY5Y cells and also in mouse cerebellum. This is an endogenous immunoprecipitation. And we identified by LC-MS/MS a few specific saxin interactors. And here I'm showing you the string enrichment analysis that disclose that the vast majority of saxin interactors are proteins belonging to the cytoskeleton. And interestingly, we also found proteins involved in organellar movement. So our hypothesis is that saxin acts as a scaffold for cytoskeleton. And we hypothesize that this intermediate filament remodeling could lead to impaired trafficking of mitochondria and also possibly of other organelles to the periphery, leading to deregulated calcium homeostasis. And actually, this is the case because when we measured the mitochondrial volume in the dendrites of primary Purkinje cells, we appreciated a striking reduction in the knockout cells compared to controls, and highlighting the fact that mitochondria are retained in the cell soma. But this impairment in mitochondrial trafficking leaves mitochondrial ultrastructure and functionality unaffected. As you can see here, indeed, the mitochondria in vivo in the cerebellum are pretty well preserved, at least they are normal. And also by measuring the mitochondrial ATP in cerebellum, this was resulted unchanged between wild type and knockout. And this is also confirmed ex vivo in the primary Purkinje cells, which display a normal mitochondrial membrane potential measured by TMRM. Interestingly, in addition to impaired mitochondrial trafficking, we observe a reduced trafficking of ER to the distal dendrites of Purkinje cell. This is the quantification of the volumetric analysis. So altogether, these results support a possible deregulation of calcium in the periphery. And actually, in the Purkinje cells, despite the basal level of calcium were preserved in the knockout cells, upon stimulation with KCL, we found that the peaks in the calcium peaks in the knockout cells were highly increased. Also in vivo data support this calcium deregulation. We perform LFQ proteomics in the Sachs knockout mice that revealed the deregulation of many proteins involved in calcium homeostasis, so calcium ion transport and chemical synaptic transmission. Furthermore, we demonstrated that the levels of phosphorylated kinase 2 beta are increased in the knockout compared to the wild type, and the levels of IP3 receptors are instead reduced. So this data gave us the rationale to test also in this model ceftriaxone. In this case, we performed a post-symptomatic trial at 5 months of age. This is the wild-type mouse on the beam, balance beam. This is the vehicle-treated knockout mouse that you can clearly see displays and very severe phenotype with many missteps and many falls. But when the mouse is treated with the drug, it shows a marked improvement of the motor performances, both in females and in males, and both in terms of latency to cross and midfoot missteps. And this rescue of the motor performances is accompanied by a delay and arrest in Purkinje cell degeneration, and also this treatment likely improves the calcium homeostasis because upon ceftriaxone treatment we saw a reduction in the phosphorylation of kinase 2 beta. So the last example in the last 2 minutes of my talk is related to another form of ARCA, which is ARCA2, which is caused by mutation in CoQ8A gene, which is an atypical kinase, which is part of a large complex that regulates CoQ10 biosynthesis and thus the efficiency of the respiratory chain. Patient fibroblasts show reduced level of CoQ10 and increased sensitivity to oxidative stress. A couple of years ago, the group of Hélène Puccio generated and characterized the knockout mouse model for CoQ8 for ARCA2, which again recapitulates feature of ARCA2 patients showing decrease in in-grip coordination on the beam test, exercise intolerance, and dark degeneration again of the Purkinje neurons with mitochondrial alteration. And since we have an ongoing collaboration, Hélène Puccio allowed me to show her recent data that support again a mitochondrial calcium connection also in ARCA2, because in the primary Purkinje cells she observed that the membrane potential was strikingly affected by CoQ8A absence, and in parallel this results in an elevation of pathological elevation of calcium level measured by Calbright. And also further supporting the regulation of calcium homeostasis, she found altered glutamatergic signaling and the same increased phosphorylation of CAMK2 and the reduction of IP3 receptor that we saw also in the RSAX mice. So in conclusion, the data that I showed you identify a mitochondrial calcium axis as a potential convergent pathway for many ARCAs, and also we propose that ceftriaxone could be a drug to be tested in all these preclinical models. So thank you for your attention. And these are the acknowledgements, and I'm happy to take questions. Thank you.
Thank you very much, Dr. Malteca, for your nice presentation and driving us into the genetics of ataxia. I have a general question. These knockout models are very potent and allowed us to see a lot of molecular aspects and controls. But all of these signatures, molecular signature, proteomic signature that you show, what are the parallels that we see in humans?
The parallel, you mean?
Yeah, I mean, you were saying that there is any indication about mitochondria sufferance and calcium changes in patients.
Yeah, of course, it's very hard to have post-mortem samples from patients, but the fibroblast data support the same mitochondrial dysfunction. For instance, generally talking about mitochondrial diseases, when there is a disease which is primarily caused by mitochondrial dysfunction, even if the disease is mostly neuromuscular, you are able, in most cases, to detect also a phenotype in the fibroblasts. Okay, and this is actually the case in the forms that we have studied. On the other hand, the aim is also to, let's say, confirm this data in the cerebellar organoids derived from the iPSCs. But I have to say that we are a bit limited in this regard because the level of maturation that these Purkinje cells derived from iPSCs can reach is very limited. So we have an ongoing collaboration with Esther Becker in this regard. So for neurodevelopmental disorder, they, at the moment, for the state of the art, they are okay. But for the neurodegenerative disorder, I think they do not express exactly the marker of a mature for Purkinje cells. So we are not, at the moment, we are not ready to do this kind of comparisons in cerebellar organoids, human cerebellar organoids.
Thank you. We have a question from Mario Manto that is asking, what about the NRF2 activators, such as homovalenoxone in NRF2 upregulation, a potentially viable solution, antioxidant response to restore mitochondria culture homeostasis? Do you think that this could be
yeah, of course, this is, can I? Sorry. Of course, this drug is now in clinical trial for Friedreich's ataxia, and it is known to stimulate the biogenesis of mitochondria and the antioxidant response. For sure, this drug could be useful, for example, in SPG7-AFG3L2 and maybe in the CoQ10, because the disease is primarily mitochondrial. And on the other hand, in the SRCA disease, our studies reveal that the mitochondrial impairment is a secondary to a cytoskeletal impairment. And so the main feature that we see is the mitochondrial, defective mitochondrial transport. So in the case of SRCA, I'm not sure that increasing biogenesis of or antioxidant activity could be successful, because mitochondria are fine, so simply they are not transported. On the other hand, maybe this can partially rescue the phenotype of the AVFG3L2 SPG7 ataxias. Even I have to say, even though I have to say these are housekeeping genes that and their deficiency causes pleiotropic, let's say, phenotype in mitochondria. So these stimulators of biogenesis could, in principle, in my opinion, let's say, improve some aspects of the disease. But as I showed you in the EFG3L2 SPG7 ataxia, there is let's say a sequence of events. So fragmentation, hotspot dysfunction, ROS production, proteotoxicity. So I'm not sure that the drug could recover all of them, but of course it might ameliorate the phenotype.
We have
I hope that I answered to your question.
I think so. We have now a couple of other questions from the audience. The first is, what are the differences between cerebellar disorders and Parkinson's disease in terms of homeostasis of mitochondria?
So it's hard to answer to this question because also in Parkinson, mitochondrial, the regulation of mitostasis is one of the most relevant themes. So probably, so there are some overlapping themes because the quality control of mitochondria is altered in many genetic forms of Parkinsonism. For instance, PINK1 and Parkin are involved in mitochondrial autophagy. So, of course, there is a connection. I think that there are a subclass of neurons that are specifically susceptible to mitochondrial dysfunction. So, for instance, Purkinje cells and maybe the neurons of the substantia nigra, and also motor neurons. So there are some neurons that for their metabolism and probably for their cytoarchitecture are more vulnerable to mitochondrial insults. So yes, there is a strong connection, of course.
Dr. Malteca, Sofia from the audience is giving a lot of compliment to your presentation and is asking if you have tried longer-term treatment. Does the drug rescue Purkinje cell degeneration at advanced stage of disease?
Okay, this is a very good question. Actually, we obtained a European grant very recently, exactly to address this point. So we are using this mouse models that I have showed you in order to try a prolonged treatment with ceftriaxone in order to see whether it is well tolerated in the long term and if it triggers toxicity. But we will also try to stop the treatment and see whether the rescue is maintained after a washout. So we are trying to test all the combination in order to translate to humans, possibly, of course.
Thank you, Dr. Malteca. I see if there are no more questions from the panelists. I don't see any raised hand. I thank you all for your presentation. Now we have a 15-minute break and we'll be back at 4:30 for the second session.
Okay, welcome back, everybody. So Jeremy Schmarman here. I'll have the pleasure of chairing this session for the hour. Hopefully people have had enough to listen to and see the lectures. I did, and they're all fabulous. And so what we thought we would do in the hour that we have, rather than just open it up and say, let's have questions fly, is have the speakers just give a little thumbnail presentation of their work as an introduction and a reminder for the session. So two of our speakers are not going to be able to join us, Drs. Ferrucci and Naylor. And I think that what we could do, I'm going to just take a moment to discuss, at least open discussion for those. If anybody had any questions, anybody who wanted to talk about the talks with each other, and then we could send questions that could come back, I guess, maybe tomorrow or in the chat to Drs. Ferrucci and Naylor. So Dr. Ferrucci, who's in Milan, gave a talk on cerebellar transcranial direct current stimulation. which is interesting and for use in neuropsychiatric disease potentially. And I wonder if anybody would like to make any comments about the talk. I know that Dr. Sherrod, if you're still on the line, if you wanted to make it, if you have any thoughts about TDCS versus TMS, how the two compare with each other, and a question that applies both to TDCS and to TMS is one that I think is sort of interesting and perhaps central is, how do we know that we're getting the cortex versus the nuclei? Are we stimulating for excitation or inhibition? I think these are some of the questions that come out of the brain stimulation of cerebellum, given the nature of the intervention that we're offering here. So does anybody have any thoughts or comments just to chat about brain stimulation, TDCS versus TMS in the few minutes that we have?
Well,
Rachel, do you have any thoughts about that?
I think this is Marco Molinari's field, but without knowing very much about the technicalities of TCDS, the impression I get is that it's actually less focal. And so probably in that situation, you'd be stimulating both the nuclei and the cortex. The thing I feel about the magnetic stimulation is it decreases with distance from the coil. So the cortex is going to get significantly more stimulation than the deep nuclei. And so while I haven't necessarily been able to prove it because I'm not an electrophysiologist, I think that you always have to hold in mind that you're likely to be stimulating an inhibitory neuron, and therefore the effects will move downstream with that situation. So if you're stimulating Purkinje cells, you're going to be inhibiting the DCN. So do you actually need to be inhibiting the Purkinje cells? And I haven't done the acute experiments, and in fact that's something that would be worth doing, is actually acutely to be stimulating during a task and see whether you need to have something that is said to be inhibitory or something that is said to be excitatory, because one tends to think of this stimulation generally and what you do to the cerebral cortex, which is much more excitatory than the cerebellum, whose projection neurons are inhibitory, I think is fundamental. And we would need to do complicated stimulation while during a task.
Right, right. Marco, do you have any thoughts about that?
Is he there?
You know, one of the thoughts that is relevant for brain stimulation here is just the very low level issue of the structure. So you have the occipital lobe sitting right over the anterior lobe, which is motor, and then you have the posterior cerebellum, which is more accessible. So do you and the trials of stimulation for the ataxia themselves have been a little disappointing, I think. Do you think that this is simply a matter of where you can reach, or is there something more deep-seated about the nature of brain stimulation and the disease in themselves? Maybe the neurons are gone. There's nothing left to stimulate.
I think that is one of the things that is fairly key. There was a paper came out of Jennifer Rogers' lab looking at, in fact, optic nerve regeneration after a transection, and they found that RTMS didn't do anything. And I think there is a degree of lesion that this magnetic stimulation can't repair. And we have had very successful results in terms of axon outgrowth and dendritic branching. We haven't actually tried to keep cells alive. We can kill them very effectively. If you stimulate for too long continuously, you raise the ROS and you can kill them. But we haven't actually tried to keep cells alive. And am fearful that if you've got a spinocerebellar ataxia with massive loss of Purkinje cells, brain stimulation is not the way to go. It's too late.
Jeremy, this is Harry. If I could jump in on your point.
Sure.
I think it's a really important issue because the imaging studies in human SEA patients have shown that at the time of clinical diagnosis, i.e. motor deficits, their cerebellum is pretty well gone through the extent of its atrophy. And I think in terms of approaches that are going to require, you know, a substantial population of functional neurons to be effective, we're going to have to go much earlier, pre-symptomatic, ideally.
Yes, I would agree with that.
Okay, thank you. Well, in a lot of it, this is a vigorous topic for research as we go forwards.
So Jeremy, one more point.
Yep.
To everybody who's participating, if Jeremy doesn't do a good job chairing this session, I have a nice picture of him from a meeting at Las Vegas that I could share with everybody.
I think that's called blackmail.
I think you piqued our interest there. All right, now Dr. Naylor, who gave the second talk, is down in Australia, and it's some awful hour of the early morning there, so he's not actually joining us. But Esther Becker is one of his co-authors, and Esther, I don't actually see your name here, but I imagine you may be here. He gave a lovely talk about cerebellar organoids, which I thought was quite remarkable, and showing that you could identifying the different cell types within the different organoids. Is there anybody who knows this work and wants to comment on it? Since Dr. Naylor couldn't join us with us. I don't see all the participants. I'm not sure if Esther's on the line. All right. Well, if you have any questions, I see the question I had about this is, and it has to do with the talk on the IPS generation as well. I remember my very first IBRO meeting when I heard Dr. Konstantin Sotelo talk about brain transplantation, and the issue that they kept hitting up against was the inability to get the transplanted tissue to send axons down into the deep nuclei. And that was a limitation to transplantation. So I was just wondering whether anybody had any thoughts about this business with organoids, whether that's only for research, does it translate to potential transplantation? Is there a way to make contact with the nuclei to actually allow the circuits to function? Is there anybody who wants to make any comments about that, to provide some insights from our esteemed scientific collaborators around the world? John, you're on mute.
I've not been using it. I say another use is from the pharmaceutical industry to test drugs on a high level, more than
great.
I don't know if it has any up to now any success, but I think this is one of the big future of this organoid, because they are arrested in development. Of course, they have many limitations, at least for me, but they can use it to do mass drug selection or things like that. Supposedly.
Is that something that, from Dr. Malteka's talk, is that something that could be used, for example, to study a disease like RSX and see whether ceftriaxone would be effective in that kind of a model? I mean, is that the kind of idea that you had in mind, John? Is that where this is going?
Sorry, I have a comment on the iPSCs, if I can.
Please.
Yes.
Yeah, because actually, it's a shame that is not here, but we are actively collaborating. So as I was saying during my talk, so the main limitation that we see at the moment is that the Purkinje neurons in these organoids are very immature, as I was saying. So she did single cell RNA-seq, but so many markers of mature Purkinje cells are not expressed. So I think that for degenerative diseases, this is a major limitation at the moment. We are trying to do organoids from SRCA iPSCs, and we will see whether we can model some of the phenotypes that we have seen in the primary Purkinje cells from the mouse. So at the moment, this is what I can add on this part. For the second, sorry, can you repeat the question?
It was about using these organelles as Jean was talking about, for looking at the response to a therapeutic intervention as a model for a drug target.
Industry.
Yeah, so the problem is the same, I think. So we first should test that the pathogenetic cascade that we see in the primary neurons from the mouse is evident also in the Purkinje cells that are derived from the iPSCs. For the moment, we don't know. Of course, if, for example, for RSACs, if we see accumulation of neurofilament and defective mitochondrial transport, I think this is sufficient to try to test ceftriaxone in the organoids.
Definitely.
At the moment, I cannot.
But I think there is also a kind of sociological problem. Pharmaceutical industry is facing the action of activists against in vivo studies. And so they push forward the idea that the organoid will solve the problem. But I don't know if this is just a way to avoid and to decrease the level of action of activists or not. I think maybe it's better for neurodevelopmental studies or pharmaceutical intervention, because clearly the organoid remain very immature now. It's very
well, let's follow that direction and see where that goes. So well, next
Jeremy,
sorry, yes, Hassan.
Yeah, well, I want to follow the Jean, in fact, comment. The neurodevelopmental guys should comment of this one. I think, well, if we put the neuron or the organoid culture, anything that we put in our tissue, they will grow. The neuron, they will, in fact, form, or we can stain, particularly calbendin for Purkinje cell. But the important thing is that the connectivity means that our Purkinje cell cannot send the axon to the cerebellar nuclei. And I presented, and I will discuss more about this one, that already this connectivity, the main one, the foundation, they should form in early development. Then interneuron, they will form or follow this foundation later on, such as the Garner cell migration or governing interneuron migration. And this way, in fact, the connectivity form. So now in the adult, you put the neuron in our nervous system. So for sure, because of we have in fact, the background and some context are IPS, they will in fact grow. But if they can send the axon in the cerebellar nuclei, probably not. With this knowledge and data we have, probably not going to work. We need lots of the work to find this one, that our axon, that they can find the cerebellar nuclei, because the foundation of their connectivity probably start in the cerebellar nuclei. Then the Purkinje cell follow this, in fact, connectivity. I presented, and I will discuss more about this one tomorrow.
Thank you. This could be a topic of a whole morning, seminar. We're going to move on.
Just a small comment.
Time is tight. We have to move on because we have 10 talks to do in the hour. So next is Harry Orr. Harry, do you have a slide that you want to just summarize your wonderful talk on CCK?
Yeah, I have a couple of slides, so I will be quick if I can. So just to remind everybody, as you alluded to, the talk that I recorded is some of our recent work on the role of cholecystokinin and its stimulation of the cholecystokinin 1 receptor, CCK1 receptor, as a way of being protective, a protective pathway, if you will, in Purkinje cells. And this work was done in collaboration with Stefan Pulse, and we used both SEA1 and SEA2 mice. And our hypothesis at this point is that the protective effects of this pathway pass through the mTOR C1 signaling pathway. And this whole work started, we had developed this transgenic mouse model that had full-blown ataxia, but did not show progressive loss of Purkinje cells. When we did an RNA-seq experiment, we found that a gene that was really dramatically increased in these non-progressive ataxic mice was CCK, cholecystokinin. We then went on and crossed these mice to CCK knockout mouse, as well as a CK.
We lost your audio, Harry.
Yes.
Harry, we lost your audio somehow. I think on your computer, you're not on mute on Zoom. Somehow your audio is going on your computer.
Oh dear.
You're back.
I don't know what.
Got it. No, you're back.
Am I back?
Yeah.
My internet is unstable. So the bottom line is that in SCA1, the mTOR pathway is downregulated, and in SCA2 in the cerebellum, the mTOR pathway is activated. And this fits in line with some work from Englagor et al. In the European Journal of Neuroscience in 2015, showing that either too much or too little mTOR signaling was detrimental to the function and viability of Purkinje cells. But the interesting thing is that in both cases, activation of the CCK1 receptor with an agonist restored proper mTOR signaling and was protective to the disease in both of these models. And this work is a preprint is on bioarchives. It's shown here. And lastly, I'll just end quickly with, we're intrigued with this pathway may be a protective pathway in other regions of the central nervous system, because we find in our SCA1 knock-in mice, where we're expressing the mutant gene throughout the CNS, a similar decrease in CCK expression in other regions. Here is the cortex, hippocampus, and striatum. So that's it, other than that this pathway may also be implicated in other neurodegenerative diseases, in this case, a paper from Bev Davidson's lab on Huntington's. So
I missed, because there was a problem with your talk. I missed the way you stimulate the cholecystokinin receptor. Sorry.
Yeah, well, actually, Jan, I mean, Gene, I didn't really go into that specifics. We use this agonist. It's called A71623. It's a tetrapeptide mimic. It shows a 1,000-fold increase in sensitivity over CCK1 versus CCK2 receptor.
Thanks.
Go to the bioarchives. It's in there.
Okay.
You know, as a systems neuroscientist who knows nothing about this stuff, it's so elegant that you found relative preservation of neurons in your mouse model. You then went to see what the gene was that was responsible for that, and then you went and you targeted that. I mean, that's so impressive.
Thank you. I think Jeremy's making a good argument for me not showing the picture that I have of him.
So are there drugs that currently promote CCK function?
Yeah, this is one of the exciting things about this, is that this pathway has been implicated in obesity and in both peripheral aspects of obesity and satiation, as well as central aspects. And I have a feeling my internet just went out again.
We can hear you. We can hear you. Are there drugs that promote CCK, or can you give a CCK analog?
There are drugs that there are both CCK1 receptor as well as CCK2 receptor agonists. So yeah, in that sense, we can stimulate the pathway. Whether we can actually promote the expression of CCK I'm not aware of any drugs in that case, but that is something we're very interested in pursuing. Is what's the pathway that elevated CCK in this one mouse that didn't show progressive Purkinje cell loss?
One more question, anybody?
Well, a small question. mTOR is also involved in longevity. How do you manage to reconcile the effect of CCK on SCA models and the effect of mTOR on longevity? Is there a link, or it has nothing to do?
Yeah, I think our experiments were so short in their duration, going beyond our target effects in the cerebellum and Purkinje cells. I just don't think I have any basis to comment on that.
I mean, the downstream pathway, the pathway are the same, probably, no?
Yeah, it depends. I think the pathways differ between CCK1 receptor stimulation versus CCK2. And we'll just have to wait and see which of those pathways are involved in various aspects of CCK stimulation.
Fascinating stuff. Thank you. We'll stay tuned for that next year for the follow-up.
Next year.
Yep. All right. So we're on to Michael Strupp, who is going to tell us about some new therapies for the ataxias. Michael, go ahead.
Yeah, I will use you. Can you see my screen?
We're about to.
Not yet. Yes.
Yeah. Okay. So during my short presentation, I have focused on the pharmacotherapy of two types of lysosomal storage diseases with the leading symptom of ataxia, Niemann-Pick type C, and Tay-Sachs and Santoff disease. And I also presented the results of a clinical trial we just finished in episodic ataxia type 2. So these are the three major molecules, small molecules we have been interested in over the last 10 to 20 years: acetyl-L-leucine, 4-aminopyridine, and an activator of small calcium-activated potassium channels, chlorsoxazone. With acetyl-L-leucine, we have published quite a lot over the last 10 years, actually. And we have a wonderful collaboration with colleagues from the Department of Pharmacology and Biochemistry at the University of Oxford. They have the equivalent animal models of Santoff disease and also Niemann-Pick type C. And this has allowed us to confirm our clinical findings by back translational research in these animal models. And we have now also a detailed idea about the mechanisms of action in these animal models. And I think the findings in terms of acetyl-leucine, we have to be very careful. We have finished a trial with more than 150 patients in various spinocerebellar ataxia. There was no benefit, but we have seen convincing effect also in a controlled trial in Niemann-Pick type C and gangliosidosis. And the drug also has an impact in restless leg syndrome, for instance, by normalizing membrane potential. And the paper came out in Science Letters by Nature that it has an impact on traumatic brain injury. This further elucidates the mechanism of action. Another study which we have just finished was a 3-period crossover trial in episodic ataxia type 2. So we compared the effects of famotidine, which is the prolonged release form of 4-aminopyridine, and acetozolamide. So both drugs were effective. However, famotidine has significantly less side effects, and therefore it was preferred by the patients. If it's not effective, you can give acetozolamide, and we have a lot of patients who use a combination of both. And here you see a spectrum of potential indications for this potassium channel blocker 4-aminopyridine. We have convincing evidence in downbeat nystagmus, episodic ataxia, but also in cerebellar central positioning nystagmus. And in my presentation, you can see a lot of videos of animal models, a mice race, for instance, but also a lot of videos of patients with various lysosomal storage diseases, which underline the clinically relevance of these findings.
Nice.
Very nice, Michael. Questions, anybody?
What is the agenda to get this potentially active drug in clinics?
Okay, so we have finished a phase 2b trial. Now we are in negotiation with the FDA for a phase 3 trial in primarily Niemann-Pick type C. And I think we are all facing, every clinician, Jeremy, I think has the same issues, a problem, and this is a clinically meaningful endpoint for clinical trials in ataxias. And this is something we're also going to discuss with the FDA. And I think this could be a hot topic to have a joint venture to work on, as I said, clinically meaningful endpoints, reliable endpoints for clinical trials in ataxia. And so far, there's no consensus about that. And that's a major methodological issue. And I think many trials failed because we do not have good endpoints.
Both primary and secondary endpoints. I mean,
well, we have the SARA, we have the SCAFI, we have elaborated a new video-based endpoint. We published that this year in trials. It's the 8-meter walk test and the 9-hole pectoral test, which is evaluated by blinded raters, the whole performance on a 0 ± 3 scale, but this was not in favor of the FDA in the end. But anyway, we have to work on that very hard and to modify the SARA. What do you think, Jeremy?
I think that the FDA is currently in deep hot water because of aducanumab. They've lost their way, and they have lost their way starting with ataxia because they don't understand it. They've not seen patients, they don't know what they're talking about, and they have the power, unfortunately. So I can go off on a tangent about this. I think the prom ataxia may be helpful because it's patient report, and the prom ataxia correlates well with the different rating scales. So that may be helpful as we go. And you know, formimipyridine is a life-changing medication for patients with downbeat nystagmus.
Absolutely.
And getting it approved by the insurance companies is a real bear. So
yeah,
I hope you'll come up with a study, and it'll be approved, and it'll make a difference to patients.
Yeah, and the same is true for episodic ataxia, because the patients benefit between the attacks. So it's not only preventing the attacks, but it improves the symptoms. And this we have seen in several patients. And our clinical approach is 4-week treatment with fampiridine, 20 milligram per day, and then washout period to evaluate the efficacy. Well, the same is true with acetyl-D-L-leucine.
Keep going, because it makes a difference, and hopefully patients will be able to benefit. So thank you for your work, and people please take a look at Michael's presentation. Okay, we're going to move on to the next one, the first to end the first half of this group, and this is Dr. Scholik from the Mayo Clinic in Jacksonville, talking about the biomarker studies in SCA3. Are you on the line with us? I'm not sure if he's on.
Yes,
There
yes, I am. Yes.
Go ahead.
Thank you for your introduction. So I have a little slide you asked me to present. So do you see that slide?
Yes, we did.
So we are a new people on the block of researching in ataxia. We here at Mayo Clinic, Florida, established the ataxia program just not even 4 years ago. And our main focus is on studying biomarkers for ataxic conditions, particularly on SCA3. And my talk today will present to you our first report. Will be probably several of them, but this is our first report that we present based on the collection of patients and biospecimens that we are able to establish over the last 4 years. As you see, we collect many biospecimens. We are particularly proud of our CSF collection, which is substantial for such a short period of time. we are actually entering the second phase of our study, that is a longitudinal project. And I will talk a little bit during my talk as well. And of course, we have a very active brain bank here in Florida with more than 5,000 brains from different conditions. So we are also recruiting these patients to our brain bank, and we already collected from SRCA cases together with our collaborators, a few of them. So you will hear our study that is done in our place by my colleague, basic scientist, Dr. Petruccelli, and his team. So I'm on clinical side of this work, and he is on the basic science work. So please start my lecture.
Thank you. Any questions for Dr. Zolek? Actually, do we know whether the ataxin-3 levels that you're measuring correlate with either disease stage or severity of ataxia? Or what are the correlations of the levels of ataxia that you're finding within the SRCA population themselves?
Well, that will be part of my talk, but the short answer is that we do not we have correlation with number of repeats, but we don't have good correlation with the clinical instruments that we use. Maybe our collection is still too small. Maybe we need to enlarge this collection. But I agree with my colleague from a moment ago that we definitely need a better clinical scales, clinical instruments that will help us with the clinical trials. But this research that we are doing will be of assistance to the clinical trials, no doubt, albeit very limited, because what we done here is only for SRCA3, for no any other diseases. So that's very limited to this particular illness. So please play my talk.
Any other thoughts or questions for Dr. Zolek? You know, if I wear my clinical research consortium for the spinocerebellar ataxia's hat for a second, the work that you're doing collecting the biospecimens is admirable, and we congratulate you for doing it. How much interaction are you having or sharing of specimens at the national level with the data collection that's going on through the National Ataxia Foundation and the SRCA?
We are privately funded. We do not have any obligations of sharing the specimens. However, we are open to share the specimens with eligible researchers through appropriate material transfer agreements between the Mayo Clinic and eligible institutions.
Yeah, thank you. I would encourage that, because there's a national movement going on to try and pool the data as much as we can for these rare resources, and all the work. We're all working to the same goal, and so we applaud you for the work you're doing, and we encourage you to be as collaborative with the other group as you can. Thank you so much, and well done for the work. Okay, we're going to go into the next half of the group, which is the pathophysiology of the cerebellum, and the first person to kick us off is going to be Dr. Egorova, who's working with Dr. Bez Przwani, whose work I've known because of the pharmaceutical company Ataxion that became Cadent, that got bought up by Novartis, that has a drug that's now sitting on the shelf and not going anywhere for reasons I can't figure out. But this is a wonderful topic about the physiology of neurons and how they're responsive to SK channels. So please go ahead, Dr. Egorova, and give us a thumbnail sketch of your talk.
Hello everyone, and thanks for the invitation. And the first part of my lecture was about the pathogenesis of spinocerebellar ataxia type 2. And I was talking about the toxin 2 protein is involved in the mRNA metabolism, and of course the poly-Q expansion interrupts the interaction of this protein with its molecular targets, and hereby the disturbed mRNA metabolism is observed in SCA2 pathology, and so this mutation kind of exerts loss of function properties. But on the other hand, the mutant toxin 2 also exerts gain of function properties, since it's known for the accumulation of the aggregates in the cerebellar tissues from the SCA2 patients and in some of the transgenic murine models. And also it was demonstrated that mutant toxin 2 enhances the IP3-induced calcium release from the ER to the cytoplasm of the affected neurons, and thus causing the cytotoxic effects and further neurodegeneration. So the cerebellar Purkinje cells are primarily affected in SCA2, and their tonic pacemaking is crucial for the proper cerebellar functioning that is disturbed in this disorder. And in our lab, using different approaches, we have demonstrated that the Purkinje cells from our SCA258Q transgenic mice exhibit impaired firing frequency and abnormal regularity of their firing. And the second part of my lecture was about the potential approaches for the therapeutics development for SCA2. And here there is a list of these approaches that I mentioned in my lecture. And in our lab, our studies are mostly focused on the recovery of the regularity in the abnormal Purkinje cells firing via the pharmacological activation of SK channels. And here is a slide with a conclusion from my lecture, and you can see like what we did in our lab so far. And so thank you for your attention.
Very nice, thank you. We have some physiologists in our group. Anybody have any questions?
I was wondering, did you see any changes in climbing fiber inputs or responses in the models?
Thank you for your question. So we ran these experiments with harmaline that activates the neurons of inferior olive. And actually, if I can, I may share my main presentation just to talk about this. Okay, so we by harmaline we activated the inferior olive neurons, and kind of more excitation was coming by the climbing fibers to the Purkinje cell dendrites, and supposedly they had to generate more complex spikes in reply, and that's what we have obtained in our lab. So in our mutant mice, the relative generation of complex spikes was significantly less than in their wild-type litter mates. So we made this conclusion that yes, the olivocerebellar tract is disturbed in our model.
Is this just a sort of question to the group? It seems like this is a generally accepted view now that the erratic firing of the neurons is the basis of the clinical phenomenology. And then the question would be is, if you can improve the firing with these drugs, are you going to improve the symptoms, or would you actually be neuroprotective and stop the neurons from dying down the road? Well, what are people's thoughts about those kind of hypotheses?
Well, in my lab, we have demonstrated the neuroprotective effect if I can like share again.
So I think this was, if I'm not wrong, I think this was Vikram Shakatai with Hank Paulson, that when they first looked at this, that if you could protect the neurons from firing off chaotically, you would actually stop them from dying. Harry, is that understanding correct? That's or Tim,
right. Yeah, Vikram has data both from several SCA mouse models that if you can alter the physiology, you also alter the loss of Purkinje cells favorably.
Yeah.
So there does seem to be a direct connection between the firing rate and susceptibility to atrophy.
And I think, you know, we're one of the lead sites in the Biohaven troriluzole study, and the results aren't out yet. But some of our patients are not reporting that they're crashing like we would have expected them to. So there may in fact be something. If it comes back positive, I think that would be a mechanism that there may be an unexpected neuroprotective effect by improving the function of the neurons. So this is nice work and has direct clinical relevance. So thank you. Keep going.
But if you change the firing of the climbing fibers, it would certainly not be the best target, because it's both armalin and kipazine are tremorogenic. They induce tremor when they so plus other drug psychiatric manifestations. So at least we need other drugs than armalin or kipazine or these things, because when you are the climbing fibers at 10 per second, you have an intense tremor of the animal in vivo.
The work I was referring to from Vikram's lab targets the spontaneous firing rate of Purkinje cells, the basal spontaneous firing rate of Purkinje cells.
So you mean more the simple spikes, probably?
Yes, the simple spikes.
Very nice. Thank you. Excellent. Well, we're not too far behind. Let's keep going. So next, we have Dr. Hilberg from Normandy, Normandy, talking to us about anxiety and depression and vestibular system. So it was a wonderful lecture. Thank you for it. Why don't you give us a thumbnail and we have time to ask questions. Pascal, you're on.
Pascal is mute.
Yeah, I think you're muted. Can you unmute yourself? Where are you? Yeah, Pascal, you're still on mute.
Yeah. Okay.
That's good.
Is that good?
Yep.
Okay. Yes. Thank you very much. It's a great pleasure to participate to this meeting. My talk deals with cooperation of the vestibular network and the cerebellar networks in anxiety disorders and depression. In fact, I'm from Rouen, from Normandy in France. My talk is, the main topic is animal behavior, because I try to test, to measure the behavior in animals with mice, with cerebellar or vestibular disturbances. And here you can see the low-term mutant mice, and it's not really easy to test them, because they have uncontrolled behavior and disinhibition, as we can see here. So very, very difficult. And long time ago, I observed this behavior, and I postulated emotional disturbances in these mice. And then, based on the main concept developed by Dr. Schmemann, we postulated emotional dysmetria. And we obtained also similar data with vestibular mutants. We imagine a parallel with these disturbances with anxiety in humans. So this is another point of view, because here we include the cerebellar participation in anxiety with anticipation disorders. What we postulate is that alteration in sensory receptors, such as the vestibular system, or in the computational center, would alter the internal model and the representation of the internal world, and then this will disturb the influence of the projective system on sensory integration, and then provoke their behavioral anticipation with false anticipation of motor command and expected sensory feedback. These will provoke alteration of the behavioral output with altered adaptation to the environmental changes, altered temporal spatial organization, and social interaction. And this is here a reassurance of these inputs. Very briefly, the vicious circle is that the cerebellum and the vestibular systems are involved in the construction of the internal world via internal models, and these permit to act on the external world. Alteration of the sensory system or the integrative system, the cerebellum. These two systems are involved in updating the internal model. So this provokes disorganization in internal representation. And these disturbances prevent any allostatic situation. We cannot find a balance. So patients become chronically unable to anticipate events in the environment. And then the first internal feedback provokes the decision-making dysfunction that alter homeostatic processing. And patients become unable to find or reach a body balance, not only from a motor, but also physiological and behavioral point of view. This is the vicious circle here, which is represented with the integration of the cerebellum and the vestibular system. And then we conclude that anxiety disorders and depression could be, at least in part, due to alteration in internal models and self-perception involving functional cooperation between the cerebellum and the vestibular system. Thank you for your attention.
Very nice models, and well done for that line of work. Any questions?
Well, I just want, sorry to speak too much, but I just want to make a small comment that is, in some of the cerebellar mutants, the lurcher and the staggerer, there is also an abnormal endocrine response to stress. So I don't know if this is a cause or a consequence, but in terms of anxiety and things, it could play also a role. I think Jan Sandelin has also published on this kind of things.
Yes, and we try to reduce the disinhibition. We can and we block. We did two things. The first is to block corticosterone level, and these did not change behavioral disinhibition in these mice. We also observed hypertrophy of the adrenal gland, and I remember your work,
Long time ago.
A long time ago. It was in 1995, if I well remember. It was with the ACTH and corticosterone level, just yes. When we touch the mice. And we also test the effect of benzodiazepine in these mice to reduce behavioral disinhibition. We reduced it, but we observe slight effect on motor functions.
Okay.
But there are no correlation between the stress level and the cognitive functions.
Okay.
Laurie, you had a question?
Yes. Hi, Pascal. Just do you have any idea about this anxiety emotional pathway that could sustain your model?
Not really. This is a very conceptual model.
Okay. So no okay. No. So you don't know, but no thoughts or no particular target that you have in mind.
At first, with the vestibular system, with the Balaban system, it was a model developed by Balaban involving the vestibular system in anxiety disorders. And he proposed in his model as I'm looking for the name of this. Sorry, I'm disturbed. I don't remember.
There is something about the thing that stands out so strikingly in the way that you're thinking about this is the integration of sensory perception, vestibular awareness, motor function, autonomic and behavioral response. And it just, I think it gets even something like something as episodic as a panic attack, which at this point is completely unexplained. Could even be something for you to think about, these sort of episodic anxiety spells. And I have a patient who at some point I'll write up who's episodic ataxia type 2, who's the induction of motion produced a true panic episode. So the whole sort of vestibular visual interaction with an anxiety component to that. So keep going. This is really interesting stuff. And I think that the more we go, the more you'll be able to answer Laurie's question about the pathways involved in getting this going. So in the interest of time, we're going to move on. Thank you for that. We're going to move on to Dr. Bumui Lee, who's talking about the primary ciliary deficits in Fragile X. Go ahead, you're on. I think you're on. Let's unmute yourself. You're still on mute.
Hi, everyone. Do you hear me?
Yeah.
Okay. So my name is Hae-Young Lee. Actually, not Bumui Lee. Bumui Lee was my first author postdoc for the paper. Yeah. So it's okay. So thanks for inviting me, especially thanks to Dr. Jan Sandelin for inviting me. And my name is Hae-Young Lee, assistant professor from the Department of Cellular Integrative Physiology in UT Health Science Center in San Antonio, Texas. So I talked about primary cilia deficit in cerebellar Bergmann glia of the mouse model for Fragile X syndrome. And you might have heard about Fragile X syndrome, but I kind of wanted to give you the brief introduction because Fragile X syndrome is one of the single gene mutate autism spectrum disorder. 20 to 30% of the patients shows autism spectrum disorder, but most of them are 100% have an intellectual disability. And it's a serious disorder because there's no treatment for any of these disorders and for the neurodevelopmental disorder in general, as you all know. And it definitely shows a cerebellar deficit. As you can see, these are listed as the cerebellar related phenotypes. And the mouse model, which is called FMR1 knockout mice, shows and recapitulate most of these behavioral symptoms or cellular symptoms that's shown in a human patient. So why do I talk about primary cilia? Because primary cilia has been known to be very important for the neurodevelopment. And obviously, it's also very important for cerebellum neurodevelopment. And this is just one representative picture that I got from the other paper that with missing primary cilia, that it can definitely disrupt the whole cerebellum development. But not only that, the primary cilia has been also recorded as a very important for the postnatal neural development, not only the early during the uteral development. So there is some connection between this neurodevelopmental disorder FXS and the primary cilia. By the way, primary cilia is immotile, which is not moving, small 3 to 5 micron, which is a little bigger than spines. And then it residue for almost every single cells, but people has found the neural function of this primary cilia recently more. So this is one slide that I prepared. So what is our question? So our question is, does primary cilia show any deficit in cerebellum of Fragile X syndrome? The answer is yes, in mouse model. And how did we get this idea? We actually published something very recently in Bente gyrus of the hippocampus. And our lab is actually not the cerebellum lab. We're doing everything about Fragile X syndrome, and then we happen to know about this fact. So dentate gyrus shows a very distinct primary cilia loss in the Fragile X syndrome mouse model. And then we thought that, oh, it might be interesting to see the other brain region. And then we happen to know about this cerebellum, which has a neuronal granule cells. Neuronal granule neurogenular progenitor cells, which is in EGL layer in their early development. And then we wanted to see how that get deficit by primary cilia. And our answer is, as you can see here, primary cilia loss in FMRP knockout mice was somehow lobe specific. So this is something that we could actually discuss about it, but we don't know the reason why. We're only seeing this primary cilia loss mostly in the posterior region of the cerebellum. And also it was a layer specific. So we only could see this primary cilia loss in the Purkinje cell layer. And in a Purkinje cell layer, there we checked the neuron and also checked the calbindin positive Purkinje cell and also BLBP positive Bergman cells. And we only could see the primary cilia deficit in the Bergman glia. So altogether, it was very interesting, but at the end, we wanted to know, yes, so from when do we see this primary cilia deficit? And it was age dependent. So we could only see this around from postnatal day after 7. So in mouse age, postnatal day after 7 was when we started seeing this deficit. So this is what we know until now. And then the second question was, does deficit contribute to FX's pathophysiology? So answer is partially yes, and then we don't know the rest of them yet, and that's ongoing work. So we were able to ablate FMRP, which is the gene product of FMR gene, and then which is causing the Fragile X syndrome by losing FMRP. So we checked when FMRP is ablated specifically in Bergman glia using Glass-Decree mice and tamoxifen injected in P1 and P2. And then we were able to recapitulate actually all these behaviors, or all these cellular phenotypes by losing Bergman glia primary cilia and then also age dependent. So we think that FMRP in Bergman glia is causing all this deficit. So that's all we have right now. And then this is before pandemic and during the pandemic, our lab and the major persons were working here, Bomi Lee, and then now Yan is also working on the primary cilia stuff. Thank you. You guys have any?
Thank you very much. Excellent. Any questions? Any questions for Hyun?
Is perhaps I just missed it. The loss of the granule cells, is it because they can't migrate down the Bergman glia?
So actually, that's a great question, because Bergman glia is most significantly, they are involved in the EGL GMP migration into IGL, right? So we think that it more affect the EGL proliferation, and then that actually cause a thickness problem rather than migration at this moment. And then we don't know exactly the reason why. So because we didn't see the migration problem yet, but we only see the proliferation problem.
I asked the question because P10 is about the time for the start of the big migration load of immature granule cells. So I was just wondering whether it was...
Yeah, that could be the really possibility, but we were able to see this from P10, but we actually didn't really test between P7 and P10. So we're on a road on testing P8 and P9 at this moment. So we don't know when exactly this start, but we were not able to see anything until P7. So that's for sure. So we didn't really dissect if this is a problem of the migration or this is a problem with only the proliferation, but we definitely see the proliferation problem because we did the BRD injection and then checked the proliferating cells. And if you see my talk, we were able to see the very obvious reduction of the proliferation itself. And the thickness of EGL was thinner, but we were not able to see anything in IGL thickness yet. So it was not in my talk, but that explains that migration might be the problem, but mostly the proliferation might be the problem.
Okay. Thank you for that excellent work. We're going to run about 10 minutes behind, so if you all be with us. We're going to move on to the next one, which is by Dr. Mobergay from Norway, who's going to present his wonderful work on cerebellum and schizophrenia. So, Torgeir, go ahead. Are you still there? Yep.
Okay, so can you see my slides now?
Yep.
Yeah, okay, so let's get that started. Okay, so we wanted to have a look at
We don't see you, by the way.
Okay. Oh, now you see me. I think it was started by the hosts. They did it for me. That's good. So let's see. Good, go for it. Okay. So we wanted to look at the cerebellum in schizophrenia, and I think thank you for the invitation, by the way. I was very honored. I think Dr. Schmamann gave a good introduction to that earlier today, why that might be an interesting question. So what we did in our work was to try to answer two questions. So first, is the cerebellum reliably affected in schizophrenia? So we use neuroimaging to look at that. And the history up until our work was that there were some studies suggesting that, and also some studies suggesting that it might not be a big problem. Most of those were very small. So what he wanted to do was to collect a lot of patients to see if that was a robust finding or not. And we also wanted to check cerebellum morphology, see if that was associated with subclinical psychotic symptoms in adolescence when these disorders typically first present. So the quick answer to this is that yes, we do see cerebellar reductions in schizophrenia patients relative to controls. When we compared almost 1,000 schizophrenia patients to over 1,000 controls. And you see these reductions primarily in more of the cognitive regions of the cerebellum, so lobule 6, cruise 1, cruise 2. And in adolescents, when we then try to look at subclinical symptoms from different questionnaires that relate to subclinical psychotic symptom severity, we found also some regions of the cerebellum that show this association. It's also mostly in cruise 1. So using quite large samples of MRI data, we can see that there is an association with having the diagnosis of schizophrenia and also with the symptom severity in a more in a sample that covers the whole range from no symptoms to fairly severe symptoms. And there's partial overlap, especially in cruise 1. We also tested for different other brain regions, and for schizophrenia, the cerebellum turned out to be one of the most consistent and strongest. So they're not very strong effects. There's huge overlap between groups, but across brain features, they seem to be fairly strong. And also when we did use machine learning to try to predict this subclinical psychotic symptoms in adolescents, we can then find out, we were able to significantly predict a bit of that severity. And when we then looked at what kind of brain imaging features were most important, again, it was this region of the cerebellum, lobule 6, cruise 1, that turned out to be the most important. And that is the region of the cerebellum that shows functional connectivity to this frontoparietal network. So yeah, a cerebellar feature emerged the most important brain predictor of psychosis. That's the very last thing, is that we also started looking at genetics, and the quick answer here is that when we look at overlap between genes coding for or involved in schizophrenia and genes involved in determining cerebellar volume, we see an effect, not a very strong effect, but a significant effect for the cerebellum. When you compare that across other brain features, we don't see that as much. So yeah, that's it. For more details, see my talk. Thank you for attention.
Very nice. Thank you. Really wonderful work. Any questions for Dr. Mobagain?
Well, slide 5 was very impressive. The cerebellum was just...
Yeah. No, it did turn out to be, I think that's one of the strengths of this work, that we did also use quite a lot of other comparison features across the brain, and across for the schizophrenia patients, some of the cerebellar regions were on top together with the hippocampus, but among the regions showing the strongest effect size, and then especially for this adolescent sample, it really turned out to be the most important feature for trying to predict symptom severity. I was also very happy with that with that figure, with that result.
Well, these are dramatic findings, and they really fit into the notion that we have to completely reconceptualize the way we thought about cerebellum, in the sense of it's such a fundamental function. It's been there forever, and its role here in neuropsychiatry is just growing exponentially. Question for you is it sounds like, if I were to ask the question, how much of the variance of schizophrenia is cerebellar, you think it's pretty high. So do you think the cerebellum is driving the pathology in schizophrenia? Do you think that's where's the proximate cause here in terms of neuropsychiatry? Do you have a sense of that?
That's the big caveat here, because all brain imaging findings in schizophrenia effect sizes are not very high, and there's also very large heterogeneity. So I think what this points to, effects there, I think it's true, but it's not very big. So it's probably explaining a bit, and there are. My guess is that there are many paths to the explaining schizophrenia, and there is some recent lesion network mapping work from Fox's group that is really interesting in that sense. And when they try to look at different brain lesions that cause hallucinations, the connectivity of those regions, which are scattered all over the brain, cerebellum, but many other regions as well. When they look at the overlap connectivity, they do find actually cerebellar regions. So I think that's the kind of work we need. We need to look at the broader networks, and in that the cerebellum might be a very crucial player. That's my guess.
Michael, you had a question?
Yeah, as a clinician, did you have a look at the impact of drug treatment with neuroleptics, benzodiazepines, which have a lot of effects on the brain and also the mobility of the patients? So did you do a factor analysis for these co-finding
very good question.
aspects?
We did. Very good question. And we did try to address at least a few of those in both studies. So we tried using, so for instance, we redid the analysis of only on the patients that did not show any evidence for problematic alcohol use. That's one thing, and also medication. Yeah, so only the drug naive, and we did see the same pattern across. So that did not explain it. That doesn't mean that there might be other confounds that we haven't tested for that could explain it. But we did see that this effect was fairly constant across the age range. So it was present also in the youngest patients, and it didn't seem to escalate. So we think it, especially in combination with these overlapping findings in youth, we think this suggests that's more of a neurodevelopmental effect than an effect of all the other things that comes with having a psychotic disorder.
Thank you.
Very nice. We should probably move on because we're already running late, and we'll finish off the session. appropriately to the Charles University in the Czech Republic, which is where our fearless leader of the session comes from, Dr. Senderin, but this is by Dr. Tichanek, psychiatric-like impairments in mouse models of the spinocerebellar ataxia. So please tell us about your story.
Okay, good evening. Thank you very much for the opportunity to present our work here. So I will try to be extremely brief. As you all probably know, spinocerebellar ataxias are very frequently associated with numerous neuropsychiatric issues. In the present work, or in my presentation, I'm showing our work where we identified very analogous behavior deficits in spinocerebellar ataxia 1 mouse model, such as anxiety and depressive-like behavior, sharply impaired cognitive flexibility, or reduced prepulse inhibition. Surprisingly, we found all these behavior impairments even at the very early stages of the disease, even before occurrence of a motor deficit and a cerebral degeneration. And finally, we found that some of these behavior abnormalities correlates directly with the hippocampal impairments that we found. So we concluded it may suggest that the psychiatric issues in the spinocerebellar ataxia could have, or at least SCA1, could have a biological basis, which may be at least partially independent of the cerebellum and ataxia. And hippocampus may be potentially interesting target for the future research of psychiatric impairments in spinocerebellar ataxias. Moreover, I also try to synthesize our results with extremely interesting results from another labs. They compared different mouse models, and they found that the mouse models with cerebral restricted SCA1 neuropathology had very different profile, behavior profile. So compared to the complex model, these mice didn't show any signs of anxiety or depression, and they showed rather opposite. They showed a reduced sign of anxiety and disinhibition, and they also showed that some of the deficit may be at least partially caused by partial loss of ataxin 1 function, which may have interesting implications for the therapy of the psychiatric impairments in spinocerebellar ataxia. Yeah, thank you very much for your attention.
Thank you. Thank you so much. Any questions for Dr. Tichanek? Tim?
That was very interesting. I think it opens this big question, and I'd be also interested in hearing your thoughts as well as Jeremy's. How do we, in the human disorders, sort out the contribution of the cerebellum from a disease effect on other parts of the nervous system? And it's really a fundamental question I think you're raising.
Okay.
Philippe, you want to start?
Okay, you can start if you want.
Okay, go ahead. Go ahead.
Okay.
Go ahead. Yeah.
Okay, I think that in humans it is extremely complicated. This is also one reason why mouse models can inspire us and give us a new insight into this problem. Because in humans it's complicated. I think the one way may be comparing different types of the cerebral diseases, which may differ in the extent of the contribution of the other than cerebral neuropathology. So, for example, you know that some SCAs are more cerebral specific than others, and we can somehow correlate the extent of these issues with the extent of the non-cerebral neuropathology. But yeah, but it's very difficult in humans, I think.
Yeah. So since you asked, Tim, I think it's obviously a key question, and I was careful from the outset as you know from the 98 paper, was to use focal injuries in cerebellum to address the question of what is cerebellum actually doing. So having established that we have this relationship, the question then is, in the more widespread diseases, what else do we know and how does it tie in together? So the question has sort of morphed into something different, which is what you're asking. And we know about the hippocampal cerebellar involvement, so there's connectivity in a number of different studies and different animal models. We know, for example, in the work of people here, Jae-Hong Su together with Rudy Tanzi, shows that there's the base 1 and ataxin 1 are interactive. And so there's something about the hippocampal damage and beta secretase and ataxin 1 interaction that may be relevant in Alzheimer's disease, and therefore perhaps also in SCA1 itself. And then the other side of this is, if the mice are less anxious, the ataxia mice, turns out anxiety is kind of self-protective, and a lack of anxiety, almost like an amygdalectomy, where the absence of fear becomes a loss of stranger danger. And that is, overly gullible, overly trusting, which is not necessarily good for self-protection. So it becomes it's a complex issue, and I think that the question needs to continue to be addressed, which is which part of the brain is doing what, and what are the different transforms that each part of the brain brings to the different neurobehavioral set. I think Philippe's work brings it out very nicely.
But I think I agree with you, but I think it's not only which part of the brain is doing what, it's also that it's a system in human, it's system disease in which you have to take care of each part of the brain and their interaction also. So yeah. It's not so simple.
Absolutely. Michael?
Yeah, I think the clearest model are patients who have a stroke in the cerebellum, as were chosen by Jeremy, or those who have a tumor. Any other disease, like multiple system atrophy and things like that, we can never exclude that there's an impact on the cerebellum and other brain areas. So these are the crystal clear models, and they have shown very convincing findings in terms of cognition and behavior and things like that. And there's no doubt, based on these findings, about the interaction between the cerebellum and higher cortical function. That's most convincing.
The question that Tim's getting at and that Philippe's work raises, though, is a really interesting one, which is, now that we know that, and so armed with that new knowledge, as you go back into the soup of different pathologies and different parts of the brain affected by these different diseases, how can we piece out which part is playing which role, and how can you then intervene in a therapeutic manner if you can't actually stop the gene from causing mischief? What are the opportunities for intervention as you try and understand the nature of the dynamics of the interplay of different brain systems and how they go wrong? So it's a wonderfully nuanced and complex question that we can now address armed with the knowledge we have from the focal pure cerebellar lesions. It becomes more challenging and therefore more interesting and even a lot more fun. I think that brings us to the end of our day. Our society president, do you want to close the day out from here and lead us on for tomorrow?
I should ask Jan. Jan, are you here, Jan?
Yeah, I'm here.
Okay, so I think we are late, so we can close very quickly by saying, see you tomorrow. And it was very nice. This first day was, for me, very nicely organized, and I appreciate the suggestion of Jeremy to ask to each people from the discussion to briefly summarize their work in 1 or 2 slides. I think it stimulates the questions. And so we should try to ask to the other chairmen to do that, because I don't know if everybody has time to see every talk that we are not directly given during the sessions. So at least it was very interesting for me, at least. And I am a cerebellar fan. And we can again thank Luc Niville and Jan for this organization. And we see each other tomorrow. And at the same moment, it's 1:13. Yes.
At the same moment, 13 of Central European time, same time as today. I would also like to thank all.
Nori will say a few words. Nori will say a few words about the society, I think.
Yeah. I would like to thank to all speakers and to all of you for joining the conference and for discussion. See you tomorrow. And I would just like to remind that after closing this virtual room in which we are now, the free discussion room, virtual room will be open. So if you want, you can continue enter that room and continue in free discussions.
Yeah, that's true.
Thank you and see
Very
you tomorrow.
Thank you, everybody. Take care. Bye-bye.
Thank you. See you tomorrow.